Here in mathematics, presently working with Deccan Education Societies, Willingdon College, Sangli. We are started with our third topic from second section, that is indefinite integration. And we came across our next exercise, which is special type of integrations. So we'll move towards PPT for that. Student, this is a chapter which is for the 6 mark for your batch only. Keep this in mind. Okay, we'll start with, now look at here. In last lecture we are studied about the integrations of the type 1 upon ax square plus bx plus c dx and integration of 1 upon root of ax square plus bx plus c dx. Okay, and one more type, that is nothing but integration of the type integral 1 upon a sine square x plus b cos square x plus c dx. Okay, what we are doing here? Here we are completing the square. What we are doing? Completing the square. Revise this. Okay, this you are completing the square. Here also you are completing the square. Here you are dividing by cos square x. Okay, and you know that it will be converted into standard form. These all about these three type of integrals, because next exercise we are taking on this type of integrations only. So first type is where you are completing the square and you are using the formula, which formula we are already studied. I think you are revised also. Here also you are to complete the square whatever inside the root sign, and then you are again using the formulas which we are studied in a special type. And in third type of integrations, here we'll multiply and divide by cos square, so that we'll get sec square upon the term in terms of tan. Okay, so if it is a term in terms of tan, you substitute tan x equal to t, and then you will get your rest values. Okay, this you are already discussed. Now I am revising it. Okay, now integration of the form. This is all about these three forms. Okay, now this form is there. Here we are using, here there is a sin x, means we are writing or we are using half angle formula. So cos, for cos of x is cos of 2 into x by 2. And what we substitute? We substitute tan x by 2 equal to t. Okay, then we'll get 2 into sec square x by 2. You are dividing, so you'll get half dx equal to dt. That means you will get the values in terms of t, because sec square x you are able to write in terms of tan. You are able to write this in terms of tan by using the formula tan x by 2 upon 1 plus tan square x by 2. Okay, so here also this is converted into t. This you will get as 1 minus tan square x by 2 upon 1 plus tan square x by 2. And again you are able to convert this into t, that is half angle formula. This type of integral you are using half angle formula. Okay, one more type is there, that is nothing but integral 1 upon a sine 2x plus b cos 2x plus c. Now this is the common in between these two, is you are using t for tan only, but angle is different. Here you are using tan x equal to t, and here you are using tan x by 2 equal to t. But finally, whatever the substitution you will, after that you will get the values of sine, cos, dx and all in terms of t only. Now look at here. So what substitutions you will get? You will get as dx equal to 1 upon 1 plus t square dt, sin 2x equal to 2t upon 1 plus t square, cos 2x equal to 1 minus t square upon 1 plus t square. I told you the similar way you will get here. Okay, so all these are the formulas on which we are going to study our exercise. So these are the formulas. So we'll begin with our exercise 3.2b. Now in this exercise 3.2b, you are having these type kinds of examples. Look at here. Now in this type of examples, what you will get, these are the which are in red colors that I will solve, because that type of different examples. Now this example is similar to the previous example which I am going to solve. Now this example is same as your seventh one. Then completing the square means this example is similar to the example which is here, and this one is also different because there is a coefficient for x square. So I will take that example. Okay, so these many examples we are going to solve in this exercise. So we'll begin with this, and this is the first kind of example, means this type of example you have to use directly your formula. We'll solve this example, integral 1 upon 4x square minus 3 dx. Okay, so now look at here, this example is integral 1 dx upon 2x square minus root 3 square. Okay, now if you'll get this, it is directly in your formula. The formula says that x square minus a square, x square minus a square is equal to 1 upon 2a log x minus a upon x plus a plus constant of integration. So we are using here, what are the factors here? X minus a means 2x minus root 3 and 2x plus root 3. So whatever the factor, this is a factor of this denominator, and you have to write the factor which is with negative sign for the numerator and with positive sign, that is in the denominator, and the coefficient of x, that you have to multiply by half. That is, again we know the corollary which we are studied from, that we are using it. So you will get this as 1 upon 2 root 3 log 2x minus root 3 upon 2x plus root 3 into half plus constant of integration by using the formula. Okay, if you'll simplify this, you will get 1 upon 4 root 3 log 2x minus root 3 upon 2x plus root 3 plus constant of integration. Here you are able to use the alternate method also. That alternate method is 1 upon 4, if you will take common, it becomes 1 upon x square minus 3 upon 4 dx. Okay, now if you will simplify this integral again, 1 upon 4 as it is, integral 1 upon, now look at here, this becomes x square minus root 3 upon 2 bracket square. So if it is 1 upon 2 root 3 bracket square dx, again you are able to use your formula, and by that formula you will get your values. Okay, and what is your value? Your value is 1 upon 4 as it is, look at here, into, now 2, 1 upon 2 into 1 upon a value is root 3 by 2, so you will get root 3 by 2 here, into log of, now again its factors, that is mod. Okay, that is nothing but x minus a means x minus 3 by root 3 by 2 divided by x plus root 3 by 2. And if you will take the LCM, it will get cancelled. This 2 and this 2 will get cancelled. You will get again 4 root 3 log, and you know that log plus 2 you will get there. So what is the simplification? I will simplify this also. So that simplification says that next step is 1 upon 4 into 1 upon 2 into 1 upon root 3 by 2, because 2a log, now you know that x minus root 3 by 2 upon x plus root 3 by 2 plus constant of integration. This 2 and this 2 will get cancelled. So immediately you will get 1 upon 4 root 3. Now we need to simplify the log. Log means if you will take the LCM here, you will get this as modulus of 2x minus root 3 upon 2 divided by then 2x plus root 3 upon 2, and these 2 and this 2 will get cancelled, and you will get your final answer as it is. So alternate method also this is applicable. If you don't remember the formula, you take 1 upon 4. You factorize also. As usual, by factorization also you are able to solve. So there are 3 alternative methods to solve this example. So use any method, but solve the example, you will get the same value here, that is 1 upon root 3 log mod 2x minus root 3 upon 2x plus root 3 plus constant of integration. Okay, now second example. Look at the second example. As usual, I told you that you are able to use any method. So here we'll begin with integral 1 upon 25 minus 9x square dx. Now here we'll write as this is a 9x square means 3x square, and 25 is 5x square. So you are having a formula a square minus x square. So 1 upon a square minus x square is 1 upon 2a log a plus x upon a minus x plus constant of integration. Now here 1 upon 2a, a value is here, that is 5, because your formula says that 1 upon a square minus x square. So then you will get 1 upon 2 into 5 log. Now you are having the factors 5 plus 3x, 5 minus 3x. These are the factors of whatever the integral is, into the coefficient of x. What is the coefficient of x? Coefficient of x is 3. So you'll get 1 upon 3 plus constant of integration. This 1 upon 3 is out of the log sign. So log function is up till here only. This is your log function. This is 1 upon 3. So 1 upon 3 is a constant multiple of log function plus constant of integration. So what you will get? You will get this as 1 upon 2 into 5 is 10, 10 into 3 that is 30, log of 5 plus 3x upon 5 minus 3x plus constant of integration. Okay, so these are the examples which we are discussed on our formula. These formulas we are already discussed by factorization method. Okay, now next is integral 1 upon 7 plus 2x square dx. Now this is third formula. Clear? Then the other second example is 1 upon a square minus x square, and now you are having integral of 1 upon x square plus a square. This is the third formula which we are going to use this example, in this example. Now look at here, this is nothing but x square plus a square. So I will write in a proper form. This form is 1 upon root 2x bracket square plus root 7 bracket square. Now this is in proper form. That is x square plus a square dx. You know that this is 1 upon a. So what is 1 upon a? That is 1 upon root 7. Look at here. You are having 1 upon root 7. Then tan inverse. So you have a tan inverse here. Tan inverse root 2 upon root 7. Why root 2 upon root 7? Because you are having x upon a. So what is x value? X value is root 2x upon a value is root 7. Therefore, root 2 upon root 7x into, now this tan inverse is completed, and the coefficient of x is root 2. So 1 upon root 2 plus constant of integration. And so you got here as 1 upon root 2. Now 1 upon root 7 into 1 upon root 2, because this one is function here and 1 upon root 2 is outside your tan inverse function. So you need to multiply this 1 upon root 7 and 1 upon root 2. So what you will get? You will get here as 1 upon root 14. You know that root of a into root of b is root of ab. This is for the product as well as for the division also. A upon b is nothing but root a upon root b. This you are already studied in fractions, or you can say irrational numbers. That is the most appropriate word, that you are learn this in irrational numbers, that is root of a into root of b equal to root ab. You are learn this in indices also. So anyways, this you are learn in all the whatever the algebra you are studied up till now. So you will get this as tan inverse of root 2 upon root 7 x plus constant of integration. I am able to write this as 1 upon root 14 tan inverse of, take root common, you will get 2 upon 7 into x. This root 2 upon 7 is constant multiple of x plus constant of integration. Okay, so this is your third example on third formula. Now next example, that is integral 1 upon root 3x square plus 8. I think you are able to understand what was your next formula is. So you recall it. You are already studied about root of x square plus a square. That formula we are going to use it here. So we'll start writing that formula in terms of x plus a. This is as usual. Root 3 is coefficient of x. Either you take 1 upon 3 outside, you will get 1 upon root 3 out of integral sign, or write as it is, because you know that if it is a linear function, you are able to take the coefficient of x in the denominator. So both methods are right. So you will get integral 1 upon root of root 3x bracket square plus root 8 bracket square dx. Now as usual your formula says that, look at your formula. Your formula says that integral 1 upon root of x square plus a square dx equal to log x plus root of, as it is the term you have to write down. That I told you for minus also. Okay, so I think you remember that whatever the hints I have given that time also. So this is nothing but 1 upon root 3 log. Why 1 upon root 3? Because this is a root 3 is a coefficient of x which you will get in the denominator. So to write in the denominator, we'll write properly as 1 upon root 3 log. As usual, we'll use the formula. This is your x plus root of whatever the term is given to you, that is root of root 3x square plus root of 8 bracket square. Okay, plus constant of integration. And finally, we'll write this as 1 upon root 3 log root 3x. As it is a square. So I will write 3x square plus 8 here. So this is root 3x plus root of 3x square plus 8 plus constant of integration. So we have seen the example directly based on our next formula, that is fourth formula. Okay, so now next example, that is nothing but again it is based on our fifth formula. All these formulas we are already studied. We are taking only one example based on every formula. So look at here. So this is your second example, that is sixth example you can say, for the sixth formula. So integral 1 upon root of, again we'll write this in the x square minus a square form, or we'll call this as t square minus a square form. So anyhow, because this x is not a single x, there is a multiple of x. So we'll write properly t square minus a square dt. This type of your formula you are already studied inside the root sign. Okay, we'll use that formula over here. So what is your formula? Your formula says that it is 1 upon root 2, means it is a coefficient of x, log of. I told you that whatever way you are writing that means function, the same function you will get with the plus sign. So it is root 2x plus root of root 2x bracket square minus root 5 square plus constant of integration. Now look at here. You will get this by using the formula integral 1 upon root of x square minus a square. Try to write the formula for whenever you are using it. So it is 1 upon root of x square minus a square dx equal to log x plus root x square minus a square plus constant of integration. So our final answer is 1 upon root 2 log root 2x. Now I will write this properly as 2x square minus 5 plus constant of integration. So you will get all 6 examples on your 6 different formulas. So we'll start with our next one. So next is nothing but integral root of 9 plus x upon 9 minus x. Now onwards we'll start using the combined formula of whatever we are studied. So we'll begin with this as usual. So we'll need to rationalize the function. Why we need to rationalize the function? Because inside the root sign you are having both the numbers which are not a square of anyone. So root is there. We don't want to root at least for the numerator. That means we need to simplify the numerator, because denominator root will work, because in our formula there is a root sign. So now we don't want root sign for the numerator. That means we need to rationalize the denominator so that it will be in the form of a square minus x square minus a square, or any other x square plus a square and all. So you try to convert that into our formula. For that we need a rationalization. That is the only simplest way to solve the example, or you can say simplify the function. Now we are rationalizing the denominator. You are rationalizing the denominator. That means you have to multiply and divide by 9 plus x upon 9 plus x. So if you will rationalize this, what you will get? This is 9 plus x bracket square. Its root means 9 plus x upon, now a plus b, a minus b, that is root of a square minus b square, that is 81 minus x square dx. Now this is in the form, that is nothing but we know that we are having a square minus x square in the denominator. So we'll start with this. So if you are having root a square minus x square in the denominator, that means you need to separate the two terms. So one term is with 9. So you will get this integral as 9 upon root of 81 minus x square dx plus, or I will separate out the term here itself first, so that it will be easy to find out how it is. So here what I will write integral 9 upon root of 81 minus x square plus x upon root of 81 minus x square dx. Now look at here. Now you are having two terms and integration is sum only. So sum is distributive or addition, subtraction. So you are able to distribute this, and by distributing you are getting this step, that is integral of 9 dx upon root of 81 minus x square plus integral of x dx upon root of 81 minus x square. Okay, after that your step will be, what is your next step? Next step is nothing but this is you will get as 9 into integral dx upon 9 square minus x square. Now we are start thinking about I1 and I2. This is your first integral. This is your second integral. Its addition gives you your I. Okay, now what is I? That's why I use two colors separately. Now look at here. So here you know that here you are having direct formula, that is 1 upon root of a square minus x square. And we are already used this in the previous example. And here, so here what will be the formula, because x is already there. So you know that if you substitute t for 81 minus x square, you are getting minus 2x, because minus sign is for the x square. So minus 2x you want. So you are able to write this. I am talking about I2 now. I2 is nothing but that is integral x dx upon root of 81 minus x square. So what will be the derivative? We want this in the form of f dash x upon root of fx. That form and this corollary we are already studied and we have solved the formulas also. So this is a corollary which is based on this corollary. This one is based, but we need to write in a proper form this f dash x. So what will be the derivative of this fx? That is fx is 81 minus x square. So what will be f dash of x? F dash of x you will get minus 2x square. Do you have minus 2x square? Sorry, minus 2x. Do you have minus 2x here? No. So again we'll start thinking that how to do that. Only thing is that it's a constant, so you are able to adjust it. So that will adjust here. So how to adjust here? That adjustment is nothing but it is given here directly. We want what we want, that you have to think. We want minus 2x. Okay, so if you want minus 2x, what you will do? You will write this as integral minus half into minus 2x dx upon root of 81 minus x square. Okay, now you got the minus 2x square. Now this minus half you are able to take outside. So minus half integral minus 2x dx upon root of 81 minus x square. Now this is in the final form. Here you are having minus 2x dx upon root 81 minus x square. I took here sign which is in black in color because you have plus sign here. This plus and this minus. This together, this plus and this minus together gives you minus sign in between. So this function is 9 into integral. Look at here. 9 into integral dx upon root of 9 square minus x square minus half integral minus 2x dx root of 81 minus x square. I have given you the workout of this. Why you are getting negative sign? Why you want half? Why minus 2x dx for the numerator? Okay. So, in first integral, we will use this formula, integral 1 upon root of a square minus x square dx is sin inverse x by a. That is direct formula. So, in second integral, you substitute 18 minus x square equal to t. Therefore, minus 2x dx equal to dt, and we'll get your value as 9 sin inverse x upon 9, because our a value is 9, plus c1 minus half t raised to minus half dt. Now c1 plus c2 I will write c, and finally you will get this value as 9 sin inverse x by 9 minus half 2 root t plus c, where c is c1 plus c2, and t again will replace by our function. So your final answer is 9 sin inverse x by 9 minus root 81 minus x square plus constant of integration. This completes our this example. We'll stop here. We'll continue in the next lecture. Here we are started with our exercise 3.2b. We took the examples which are based on directly the formula, and we'll stop here. Thank you. We'll continue in the next lecture.
सह्याद्री वाहिनी, प्रत्येक घरासाठी, घरातल्या प्रत्येकासाठी.
मन की बात, माननीय पंतप्रधान श्री नरेंद्र मोदी करतील मन की बात. 27 डिसेंबर 2020 रोजी सकाळी 11 वाजता माननीय प्रधानमंत्र्यांच्या प्रसारणानंतर प्रादेशिक भाषेतील अनुवाद आकाशवाणी आणि दूरदर्शनच्या संबंधित प्रादेशिक केंद्रावरून केला जाईल. सह्याद्री वाहिनीवरून मराठी अनुवाद सकाळी 11:30 प्रसारित होईल आणि पुनःप्रसारण रात्री 8 वाजता होईल.
Go 2020 Go! Unlock yourself for 2021! कोण आहेस तू? मी राधा. माझ्या किशूची राधा.
आजोबा तर माझ्यावरून फास्ट निघाले.
नवीन आले साल आज.
पाहुणा, नामी मिरची, नामी कोल्हापूरची.
मग
मी आहे डिजिटल इंडियाची.
पाहिला म्हणजे सगळ्यांची.
एक कदम स्वच्छता की ओर.
हे स्वच्छता की ओर नाही, हे तर आज स्वच्छता की ओर आहे.
डिजिटल 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 झाला हो इंडिया डिजिटल.
नवीन वर्षाचा कार्यक्रम गो 2020 गो 31 डिसेंबर 2020 रात्री 10 वाजता
पाहायला विसरू नका, फक्त सह्याद्री वाहिनीवर. गो 2020 गो.
जब तक दवाई नहीं तब तक ढिलाई नहीं.
माझे कुटुंब माझी जबाबदारी.
हम भारत को आत्मनिर्भर बनाकर रहेंगे. देश में एमएसपी भी रहेगी और किसानों को कहीं पर भी अपनी उपज बेचने की आजादी भी रहेगी. भारत पूरी दृढ़ता से देश की एक एक इंच जमीन की, देश के स्वाभिमान की रक्षा करे. भारत माता की जय. सियावर रामचंद्र की जय. सिया राम.
सीएए पास होने के बाद देश भर में भ्रम फैलाने का काम किया.
या वर्षभरातल्या महत्त्वाच्या घटनांचा आढावा घेणार आहोत दृष्टिक्षेप 2020 या 6 भागांच्या मालिकेतून. तेव्हा पाहायला विसरू नका. 26 डिसेंबर ते 31 डिसेंबर या कालावधीत रोज रात्री 8:30 ते 9:30 या वेळेत दृष्टिक्षेप 2020 फक्त सह्याद्री वाहिनीवर.
Hello student, I am Kadambini Balakrishna Patil, teacher in mathematics, presently working with Deccan Education Societies, Willingdon College, Sangli. Okay, we are started with our topic indefinite integration, in which we are started with exercise 3.2b. We saw the few examples which are based on directly the formula which we are already learned in the previous lectures. Now we have learned some few more formulas that also we'll discuss in your examples. For that we'll move towards the PPT. So our next example is integral 1 upon x square plus 8x plus 12 dx. In the last lecture we are discussed this type of examples, what to do for this type of examples. And what we have to do, we have to complete the square for the denominator. Okay, now to complete the square, we'll discuss what we have to discuss. We have to discuss the quadratic equation which you have. What is the quadratic equation? You have x square plus 8x plus 12. Okay, now middle term. Think about the middle term, that is x square plus 2 into 4 into x plus 12. Now you want 4 square here. Okay, so what is that? X square plus 2 into x. Already x is here, x square into 4 means you want 16, but you are having 12. So plus 12 minus 16. Whatever the 16 you want, that added and subtracted. So from this you will get this as x plus 4 bracket square. Now plus 12 minus 16 is nothing but minus 4. So what is the value of x square plus 8x plus 12 equal to x square plus 4 bracket square minus 16. We'll expand and we'll see that also. X square plus 8x plus 16 minus 4 is nothing but the 12. Okay. So here is your completing the square method. We'll start with that here in our example. So what is our example? Example is I equal to integral dx upon 8x square plus 8x plus 12 plus 4 minus 4. That means this also gives you 16. Either use the method which I have discussed just now by your completing the square, which you are already studied in 8th standard, that the last term equal to, we are using that last term square is equal to middle term. Okay, middle term divided by this formula. I think you know and you will remember this, middle term upon 2 into root of first term. So that also you are able to use. By that you will get immediately add and subtract this term. You will get that method I have used to explain you for the completing the square. So now look at here, by my method I know that 16. Now you are in 12th standard, so you know that what is the square of the last term. So you will get this is plus 12 plus 4 minus 4. You will get the simplification is dx upon x square plus 8x plus 16 minus 4. Now this is complete square. So this complete square gives you integral dx upon x plus 4 bracket square minus 2 square. Now we'll use the formula. Which formula you will use? That is, because this is in a proper form, that is 1 upon 2a log x minus a upon x plus a plus constant of integration. This is nothing but the linear function. So you are able to write here t. So this becomes t square minus a square directly in the formula, because if you will substitute x equal to t, x plus 4 equal to t, you know that dx equal to dt immediately. So same formula you are able to use here. So immediately you will get the here, this is a square, means you are having a square here, t square here. Now look at here. So directly you are able to write this formula as, what is your formula? Your formula says that 1 upon 2a. What is the value of a? This is a. 1 upon 2 into 2 log of, now t minus a, t plus a, because here factors are x plus 4 minus 2 and the other factor will be x plus 4 plus 2. So these two factors you will get here because it is a 2 square. So x plus a, x minus a, and this factor is on the numerator. This factor is in the denominator. So what value you will get? You will get this value as 1 upon 4, because this is 1 upon 2 into 2, 1 upon 4 log x plus 2. Why x plus 2? This is plus 4 minus 2. So you will get this as plus 2, x plus 2 upon x plus 6 plus constant of integration. Okay. So now you will get this as 1 upon 4 log x plus 2 upon x plus 6 plus constant of integration. Okay, next example. We'll start with our next example, that is integral 1 upon 4x square minus 20x plus 17. Okay, again we are using completing the square, but here there is a coefficient of x square also. So here you need to use your formula which I have discussed in the previous example. What is that? That is nothing but you want last term. So for the last term, you want to write a middle term. Middle term is minus 20x divided by 2 into root of first term, root of 4x square. What is root of 4x square? That is 2x. So x will get cancelled and 4 5s are 20. Okay, 2 2s are 4. This is 2 into 2 is 4 and 4 5s are 20. So that is minus 5. Minus 5 square because last term square we want. So square of this term is minus 5 square, which is nothing but the 25. So we are added here 25. So if you are added 25, you are subtracted also 25 plus 17, it's already there. Here we are using again by this formula, you are getting the last term. This you are already used. Okay, so now you got a perfect square for this. What is that perfect square? 2x minus 5 square. So you will get your next step as integral dx upon 2x minus 5 bracket square minus 25 plus 17 is minus 8 and minus 8 is here. Okay, so this is equal to integral dx upon root x minus 5 square, and this minus 8 I am able to write as root 8 bracket square. So now this is in our formula which we are already studied, that is x square minus a square. Why x square minus a square? This one is linear. For this, if you will substitute, you know that it is converted into t square minus a square formula. So look at here your formula. Formula says that 1 upon x square minus a square equal to 1 upon 2a log x minus a upon x plus a plus constant of integration. We are using here. This one is a. This complete is, I will use capital letter. So this is nothing but integral dx upon capital X square minus a square. So I am able to write as 1 upon 2a. What is the value of a? That is root 8. Then what? Then you are having log, mod of. What is x here? Now this is nothing but capital X. Mine capital X is 2x minus 5. So I will write 2x minus 5. Then I am having this as minus root 8. And in the denominator I will have 2x minus 5 plus root 8 plus constant of integration, but there is a coefficient of x that you will get here. So you will get 1/2. Don't forget to write that, and then you are able to write constant of integration. So here we are using this formula in this manner. So we'll use that and we'll get our answer as 1 upon 2 root a log 2x minus 5 minus root 8 upon 2x minus 5 plus root 8 into 1 by 2 plus constant of integration. Okay, now you got this constant of integration and you need to simplify. Here is 1 by 2. So 1 by 2 into 2. So you will get 4. And I will write this 8 as 1 upon 4 into 2 because 4 is a perfect square and it is like a root of a into b. I am able to write root a root b. So you will get here root 4 into root 2, but root 4 is 2 root. That you are able to write here. That's why we are simplifying, because we want more simplified answer. So you will get here as, that is nothing but this integral is equal to, now look at here, this is from here you will get, this is 4 into 2 root 2, which is nothing but 8 root 2 log. Now this is again you will get same value for root 8. We are writing for the root 8 value as 4 into 2. So which is 2 root 2. We'll write that. So you'll get 2x minus 5 minus 2 root 2 upon 2x minus 5 plus 2 root 2 plus constant of integration. Okay. So this way you need to simplify many terms. Next examples are, look at here. One is integral 1 upon 5 minus 4x minus 3x square. Then you are having the same quadratic inside the root sign, and we have discussed for that also what is the, when, what will happen, or you can say if it is a quadratic inside the root sign. So that type of example also will take and will simplify. Then you are having here examples which are based on cos square. Then you are having the example which are sin x upon sin 3x dx. Okay, so this type of examples we are going to solve. Either we'll convert this into the proper form, or you know that this formula also here you are going to use it. That is nothing but cos 2x. Cos 2x is equal to 2 cos square x minus 1. Either you use it, you are able to use this as 1 minus 2 sin square x. That is more appropriate to solve this type of examples, and immediately you will get your values by dividing and the multiplying by cos square x. That formula where. So we are using here, so we are solving 18th example. So it's easy to solve the 19th example. Out of these examples, because this is also quadratic equation, if you simplify, what you will get? Root of x minus 3 into x plus 2. Look at here. So that also gives you, what it will give you? For this I am discussing now. Root of x minus 3 into x plus 2. So if you will expand this, you will get x square minus 3x plus 2x minus 6, which is again a quadratic equation, that is x square minus x minus 6. So this is again of the type any one of this example. So we'll use this. So for that we are solving this 14th example. Okay, so we'll start with our next example, and our example is 14th one. That is nothing but integral 1 upon root of 3x square plus 5x plus 7. Here again, I have already discussed in previous lecture that you need to complete the square. So now you are having 3x square plus 5x plus 7. So to need to complete the square, now look at here. This is nothing but 3x square plus 5x plus 7. So either you take 3 common, or by using your method last term equal to, that also I have already discussed. So we'll write here. Look at here. So if you'll take 3 common, then also you will get the middle term, because as it is you will get the root 3 sign, you know that. So I have discussed this method also, that whenever there is a coefficient of x square or the coefficient of x, you take that common outside. So here if you will take 3 common, so you left with 1 upon root of 3 I will take outside. You left with x square plus 5 upon 3x plus 7 upon 3. Now it's a product. So you are able to write this as integral 1 upon root 3 into root of x square plus 5 upon 3x plus 7 upon 3 dx. Now look at here. This 1 upon root 3 is constant. So it is here. And it becomes integral dx upon root of x square plus 5 upon 3x plus 7 upon 3, which is your left factor of your root sign. So we'll begin with that. So after that, now as usual, you need to complete the square. Now so far this completing the square, middle term must be in terms of 2 into. So there is no 2 in this middle term. So how to find this 2 into middle term? That is nothing but, what is the simplification of this, that I will tell you. That is working as x square. Now you are having 5 upon 3, but we want 2. So divide by 2 into x plus 7 upon 3 as it is. First we will solve the problem of middle term. Okay, so you will get this as x square plus 2 into x into 5 upon 6, because this is a multiplication. So I will write this properly. Again you think about this. What is the working here? That is x square plus 2 into, we want everything. So we'll divide by 2. You will get 5 upon 3 into x. We are discussing only about the middle term. So now middle term is always x square plus 2ab. We say that. So 2 you want, so 2 is here, that is separately. Then you know that a value means here is the a value plus b square. So what is the a value? A value is from here. So here is the a value that gives you x into, now remaining completely you have to write as 5 upon 6 plus 7 upon 3. Now you got the b here. This is 2ab, a square plus 2ab. This value is b, that means you want square of this. So add and subtract the square of this. That is your next step. So in this next step, what we'll do here as x square plus 2 into 5 upon 6x. Now this is, look at here, 5 upon 6 square is 25 upon 36. That is added and subtracted plus 7 upon 3. Okay, so now this is complete square. So that complete square you will write as x plus 5 upon 6 square plus, now the working is plus 25 upon 36 minus. So you will get this as plus 25 upon 36, that is for the completing the square, that is 2 into x into 5 upon 6 plus 25 upon 36 minus 25 upon 36, and you are having 7 upon 3. So this simplification is needed here. So this simplification is nothing but, again we want, this is a 3. So what is the LCM here? So LCM is nothing but, part we want is that you have to subtract, and this fractional subtraction is already known to you. So we'll start with that. 7 upon 3 minus 25 upon 36. 12 3s are 36, isn't it? So you will get here, you have to multiply and divide by 12. So you will get here as, what is 36 in the denominator? Then what is 12 into 7? 12 into 7 is 84 minus 25. So 84 minus 25 gives you, that is the value which you will get as 59. Look at here, this is 59 upon 36. This is the workout for minus 25 upon 36 plus 7 upon 3. That is 25 upon 36. So what you are getting, this is nothing but previously, what will be your previous step? Previous step will be x square upon 5 upon 3x plus 7 upon 3. This is equal to x plus 5 upon 6 square and plus, here you are having 59 upon 36. That you are able to write in terms of the square term. So you are writing root 59 upon 6 square is 36. Okay. So these are the workings behind whatever you are getting immediately here. So this is x plus 5 upon 6 square plus root of 59 upon 6 bracket square. Now substitute in your integration sign. That is 1 upon root 3 integral dx upon root of x plus 5 upon 6 square plus root 59 upon 6 bracket square. As usual, you know this formula. We are used this formula. That is integral of 1 upon root of x square plus a square dx is log of mod of x plus root of x square plus a square plus constant of integration. So we are using here. So what is x? X is, this one is x. Now I'll use capital letter so that you are able to judge what is your comparison. Okay, so here your comparison is 1 upon root of x square plus a square. Why I am using capital letter? Because small letter is already used in your equation. This is nothing but it is log of dx is there, log of mod of capital X plus the expression as it is plus constant of integration. That is your formula. And in this formula, what you will write? Look at here. What is x? Means x value is log of whatever is capital X. Capital X is x plus 5 upon 6 plus this complete expression is here. So complete expression means this value. As it is this value, so any value you are able to write here. And that simplification also we'll see here, so that you will get your final answer. So we'll start with that. So 1 upon root 3, it is already there. Now log of this is x plus 5 upon 6 plus root of the function whatever inside integral sign as it is we need to add here. So that is root of x plus 5 upon 6 square plus root 59 upon 6 bracket square. This value is nothing but here. What is this value? This value is x square plus 5 upon 3x plus 7 upon 3. That value will substitute. So you will get x plus 5 upon 6 root of x square plus 5 upon 3x plus 7 upon 3. More simplification is possible here, and I think you should simplify that because that type of questions will be there in your JEE exam. In JEE exam, there is an alternate answers are also there. So if you'll take the LCM here, what you will get that we'll discuss here, and then we'll continue with the next. Okay, so here you are having 1 upon root 3. First I'll start with here itself. 1 upon root 3 log. So what you will get here 6x plus 5 upon 6. And what you will get from here, that is root of 3x square plus 5x plus 7 upon 3. This is inside the root sign plus constant of integration. And now you are able to write this function as 1 upon root 3 log of mod 6x plus 5 upon 6 plus root sign root 3x square plus 5x plus 7 upon root 3. And you are able to take the LCM here. And what is the LCM here? This 6 you are able to write as 2 into 3. And this 2 into 3 you are able to write as 2 into root 3 square. So if this is a 2 into root 3 square, your LCM is 6 here. And what you will get? You will get here 1 upon root 3 log. Now look at the simplification. It is 6x plus 5. Now 6 is your LCM. That means here you have to multiply and divide by 2 root 3. So you will get plus 2 into root 3 root of 3x square plus 5x plus 7 upon 6. And this gives you the one more answer. Then one more answer is minus log 6 will be using the log law. You will get different one. So these are the many answers which you will get by the simplification of this function. Okay. So next example. So you need to simplify, maximum simplification is possible in at least while solving the examples at home. So now next example is I equal to integral 1 upon 4 plus 3 cos square x dx. As usual, there is no substitution for the cos square x. Now you know that you are having cos square x. If you will divide by cos square x for numerator and denominator, it will be converted into sec square. That means again you need to substitute for the tan because sec square x is 1 plus tan square x. And once if you know it is a 1 plus tan square x, you know how to convert that into a substitution. So we'll begin with our example. So look at the example. Example says that this is integral 1 upon 4 plus 3 cos square x. Just now I have discussed that we need to convert this into sec square. That means we need to divide numerator and denominator by cos square x. And we have seen this type of formulas previously also. So we'll start with that. So if you will divide here, what you will get? You will get here as integral 1 upon cos square x. You are dividing to the numerator as well as you are dividing to the denominator. 4 plus 3 cos square x dx. Now look at the working which gives you this step. Okay. So here is integral 1 upon cos square is sec square. And you are dividing by the cos square means what way you will write? 4 upon cos square x. Okay, this is division for both numerator and denominator plus 3 cos square x upon cos square x dx. Now 1 cos square will get cancelled. Both this term will get cancelled. Here it is again 1 upon cos square. So you will get this as sec square x upon 4 sec square x plus 3 dx. And this sec square x, look at here. This is your step. After this much simplification, you will get this step. So you need to simplify. You will not get directly any value. Here if you are saying divide numerator and denominator, you must divide it. So you will get integral 1 upon cos square x upon 4 plus 3 cos square x upon cos square x dx. Then separate out the denominator. 1 upon cos square is sec square. And what is next step? Next step is substitute for tan square as sec square minus 1 or 1 plus tan square as sec square x. So that substitution is here. So what is the substitution? Substitution is sec square x dx as it is, 4 as it is. Then this sec square x you are able to write as 1 plus tan square x plus 3, because 3 is there. Don't forget to write that 3. So 3 is there. So you multiply by 4 to every term. So by multiplying, what you will get? Look at here. The sec square x as it is. Now there is also middle term or middle step to explain this next step. That is 4 plus 4 tan square x plus 3. So now this 4 plus 3 is 7 here. Okay. So you will get this as 4 plus 3 as 7 and 4 tan square x. So 4 tan sec square x dx upon 4 tan square x plus 7. Put tan x equal to t. Okay. When you will substitute tan x equal to t, what value you will get? You will get this value as sec square x dx equal to dt. So once if you know that this one is dt, you know that this is dt upon 4t square plus 7 again in your formula. So look at here. That gives you integral dt upon 4t square plus 7 after some substitution. So after substitution, if you will get this value, now what we'll do? We'll explain this. Now this becomes 2t square plus root 7 square. Now this is in the formula like x square plus a square. And we are already know the formula for x square plus a square in the denominator. That is tan inverse of x by a. So what value of tan inverse of x by a? That is 1 upon root 7. Actually you are having this as dt upon what it is? 2t square plus root 7 square. Look at here. So this value you have. According to that, what is the formula? Formula says that integral x square plus a square equal to 1 upon a tan inverse of x by a plus constant of integration. Write the formula every time and you will get 1 upon root 7 tan inverse 2t upon root 7. 2 is multiple to t. You will get 1/2 here. So you will get this as 1 upon, you are using this formula which is here, and you will get the final value as 1 upon 2 root 7 tan inverse 2 tan x upon root 7, because we need to substitute for t plus constant of integration. We'll stop here. In this lecture, we have seen few more examples based on exercise 3.2a and for few more types also. So we'll stop here. Thank you. We'll continue in the next lecture.
सह्याद्री वाहिनी प्रत्येक घरासाठी, घरातल्या प्रत्येकासाठी.
Hello students, this is Mrs. Shweta Ladha from Ferguson College, Pune. And we had started with our chapter 3, Inheritance and Variation. We've already discussed about monohybrid cross, dihybrid cross and the 3 laws of Mendel's. We had seen that Mendel's first law, it was not universally accepted, the law of dominance. Why? I'm going to tell you the reasons today. The second law, law of purity of gametes, it is universally accepted. The third law, law of independent assortment, is also not universally accepted. Why it is not universally accepted? We are going to discuss about it today in today's presentation. Also, we will see some work which was done post-Mendel. That work is called as neo-Mendelism, and how different experiments prove that at some places some new conclusions have been drawn which were different from Mendel. So we will be studying about neo-Mendelism today. So let us begin with our PowerPoint presentation. Hello students. So let me tell you that the third law of Mendel, law of independent assortment, is not applicable to linked genes. What are linked genes? You can see a beautiful picture on your screen. Let us look at the first picture. Suppose I will label this picture as A. In A picture, you can see that this gene G and A, these are placed very close to each other. So capital G and capital A, they are called as linked gene because they are closely placed to each other. Again in this chromosome, small g and small a, they are linked gene. What happens to these linked genes whose loci are close to each other? When crossing over takes place, you have studied meiosis, how chiasmata formation occurs in meiosis 1. You remember this prophase 1. When crossing over takes place, there is a chance that these genes, when they cross, the linked genes, when they cross, this portion of this chromosome, it breaks and it recombines with the other. You can see, I'll just call it as blue color, pink color gene so that you understand. So to this blue chromosome, the pink color has attached, and to the pink chromosome, the blue color attached. But look at the position of gene. Capital G was together with capital A. Look here, even after crossing over, capital G is with capital A and small g is with small a. Means they did not segregate with each other. They do not separate with each other and they did not assort independently. So thus, third law of Mendelism fails here. What did Mendel said? Mendel said that genes or members in the allelic pairs, they will segregate from each other and they will assort independently to form different combinations of the gametes. But you can see here, when they are linked genes, they do not segregate from one another. They continue to stay together. Suppose the genes are unlinked. Look at this picture. Here capital G and capital A, they are far away from each other. Such genes which are far away from each other, where the loci are apart from other, they are called as unlinked genes. Now when crossing over takes place, look, this portion is breaking, this portion is breaking, recombination occurs. And look over here, in the first picture, capital G, capital A were on the same chromosome, but here you can see that capital G and capital A has segregated, independently assorted. So Mendel's law is applicable only for unlinked genes. It is not applicable for linked genes. Students, I hope you understood what are linked genes. Linked genes are those genes which are closely placed to each other. The loci are close to each other and they have a tendency to stay together, to inherit together. They do not undergo segregation. They do not undergo independent assortment. So linked genes do not follow Mendel's third law. Therefore, third law of Mendel's independent assortment is not universally applicable only for linked genes. I hope you followed this concept. This diagram is self-explanatory. Let us proceed now. Now, why was Mendel so successful? He was working in a time when nothing was of chromosomes was known, gene was not known, and he gave out 3 laws. He carried out so many experiments. Why was he successful? Mendel was successful because his choice of material was wise and perfect. He has selected pea plant, which was an annual plant. He could get good crops. There were 7 pairs of contrasting characters. He was very methodical. First, what he did, always he started with pure lines. And as he started with pure lines, he got perfect results. He considered one trait at a time. Okay, then he kept qualitative word, quantitative records up to 3 generations. He maintained the records up till 3 generations. He made ample of crosses. It is said that about 2,000 to 3,000 crosses he carried out. And also he was lucky enough. Luck also played a role in his success. Why am I saying this? You'll come to know immediately. As all the 7 traits selected by him showed complete dominance. They followed the law of dominance, that the character which expressed itself in the first generation was dominant, then the other which was suppressed was recessive, and the recessive was able to express in the F2 generation. So whatever traits he selected, all the 7 traits, they showed complete dominance. So he was successful. And all combinations of allelic pair showed independent assortment. And just now I told you, linked genes, how they are not showing independent assortment, isn't it? They are continuing to stay together. They don't follow the third law. But when Mendel studied his genes, his allelic pairs, they were showing independent assortment. Now let us begin with post-Mendelism. In post-Mendelism, we are going to study many important concepts. Deviation from Mendelian ratio. After Mendel, many other scientists carried out experiments on different plants and animals, and many deviations were observed. Different gene interactions were discovered. This study is called as neo-Mendelism, or it is called as post-Mendelian genetics. It is very interesting. I am sure you are going to enjoy this. He observed that phenotypic expression of a gene can be modified or influenced by the other gene. This was observed. These genes interactions, they are of 2 types. We are going to study intergenic expressions and intragenic expressions. Let us see about them in detail. So intragenic, within the gene. And second, we are going to study intergenic, 2 different genes. So intragenic interactions, they are also known as interallelic interactions. I will explain this nicely to you. Intragenic, within the gene, or interallelic interactions occur between 2 alleles of the same genes. Suppose this is a pair of chromosome, correct? This is an allelic pair. Here some gene is there, here some gene is there. And when there is interaction between these 2 alleles of the same gene, it is called as interallelic, or it is called as intragenic. Example, here we will study about incomplete dominance. We will also study here about codominance. Interesting examples. Intergenic, which are also called as non-allelic interactions. These are interactions between alleles of different gene. For example, this is one chromosome. Here there is one gene, here there is one gene. These are alleles, and here there is another gene, and here there is another gene. If the interactions is occurring between 2 different pairs, this A gene I will label and this B gene. If the interaction is occurring between A and B gene, which are not alleles of each other, they are alleles of different genes, but they are located on either same chromosome or different chromosome, and if they cause some changes in the expression, such type of interactions will be called as intergenic, between 2 different genes. And they are not alleles, so they are non-allelic interactions. Here we have to study complementary genes, supplementary genes, epistasis, pleiotropy and lethal genes. We will be studying about pleiotropy and multiple allelism here. So let us study first about intragenic interactions. In intragenic interactions, I'll teach you about 3: incomplete dominance, codominance, and multiple alleles. These are the interactions between the same gene, okay, interallelic interactions. And second is intergenic interactions. Intergenic, that is non-allelic, occur between alleles of different genes on same or different chromosome. This is chromosome, this is chromosome. This is one gene, this is one pair of allele, and this is the second. This is suppose A, B. And if the interaction is between A and B, 2 different alleles, different genes, but they may be on the same or different chromosome, then it is intergenic. I hope you understood. Here we'll study pleiotropy, polygenes, epistasis, supplementary, complementary genes, out of which we'll be studying only pleiotropy. Let us start with incomplete dominance. Here both the genes express themselves partially. Incomplete, not complete dominance. Expression is partially. One gene cannot suppress the expression of the other completely. According to Mendel, dominant gene is going to express itself, recessive gene is going to be suppressed. But here, one gene cannot suppress the expression of other completely. There is no dominant, no recessive. Nobody is dominant, nobody is recessive. Both are showing partial expression, incomplete expression. And F1 hybrids are intermediate. They are neither like dominant parent nor like the recessive parent. They are in between, intermediate expression. It can be seen in 4 o'clock plant, Mirabilis jalapa. It can be also seen in Snapdragon, Antirrhinum majus. We'll see the wonderful example when red pure variety of Mirabilis jalapa or Antirrhinum majus is crossed with a white flower variety. In F1 hybrid, neither red is formed, neither white is formed, pink color flowers is formed. So intermediates are produced because of incomplete dominance. In F2 generation, 25% are red, 50% are pink, and 25% are red. We'll study this with the help of chart. It will become easy. Genotypic and phenotypic ratio both is 1:2:1. Let us look. So here suppose this is the parental generation. The phenotype is red color. Genotype is capital R, capital R. We are crossing it with a plant having white color flower, phenotype, genotype small r, small r. When F1 generation, it was found that according to Mendel, the flower should be red in color because red is dominant over white. But in this example of Mirabilis jalapa or Snapdragon, it was found that the flower were not red, neither white, all were different color, they were pink because both of them, they express themselves partially. When this was allowed to undergo selfing in F1 generation, when it was allowed to undergo selfing, gametes produced are capital R, small r. So suppose this is treated as a male gamete, female gamete. When capital R came together, it formed red flower. So how many red were formed? 1. When heterozygous condition capital R, small r came together, pink were formed. How many pink are formed? 2. And white flower, small r, was produced. So phenotypic ratio is 1:2:1. What was the phenotypic ratio according to the monohybrid cross? According to Mendel, it should be 3:1. But it is deviated from 3:1 to 1:2:1. And the genotypic ratio, capital R is 1. Capital R, small r, heterozygotes are 2, which are giving pink color. And small r is 1. So this is where incomplete dominance is shown by both red and white to produce pink color flower. All pink in F1 generation. And in F2 generation, the ratio obtained is red is to pink is to white is 1:2:1. So both the genotypic and phenotypic ratio obtained is same. This is incomplete dominance. I hope you understood this. Found in Snapdragon, Mirabilis, jalapa. Okay, so remember it. Practice this. Sometimes a difference is asked between incomplete and codominance. So let us see the next example, that is codominance. Co means together. Here both the genes in an allelic pair express themselves equally. There it was unequal, incomplete. Here it is equal expression. Classic example is of cattle coat color. When a cattle with a red coat is crossed with another cattle with a white coat, F1 hybrids are row 1. Now what is this row 1? I'll show you it with a chart. Very interesting. And row 1 is also a mixture of red and white coat. When you see the diagram, it will be very clear to you after seeing the picture. Thus, both colors are expressed equally. In incomplete dominance, partial expression is seen. In codominance, equal expression is seen. In F2 generation, 25% are red, 50% are row 1, and 25% are white. So, the genotypic and phenotypic ratio here also is similar to incomplete dominance. It is also 1:2:1. But way of expression is different. Let us see this beautiful example. Suppose we are considering a cow which is having a red coat, and this red coat female cow, she is allowed to mate with a bull which is white coat. Now here this is white. Why we are writing it with also capital alphabet WW? Because it is dominant in itself. But when they are allowed to undergo cross, in F1 generation you can see both the genes are expressing themselves. They are codominant. Red also is expressed and white also expressed, and so there are patches of red and white produced. Such organisms produced are called as row 1. So in F1 generation, all organisms produced will be row 1. Equal expression of red and white color gene will be occurring. When they are allowed to undergo selfing in F2 generation, again 1 complete red, 2 row 1 and 1 white. So again here it is deviation from Mendelism. According to Mendel, phenotypic ratio should be 3:1. What is the phenotypic ratio coming here? It is coming 1:2:1. And genotypic ratio is also 1:2:1. So what is the difference between incomplete and codominance? In incomplete dominance, partial expression, unequal expression, incomplete expression. In codominance, equal expression. Both the gene says we will show our color. And therefore here white is also appearing, red is also appearing, row 1 color. There red was also showing little, white also was showing little, and so little red and white became pink in color. So you should be able to understand the difference between incomplete dominance and codominance. Though the genotypic and phenotypic ratio is same, 1:2:1. Sometimes difference is asked, sometimes a short note will be asked. So this question can be asked for 3 marks. Prepare it nicely. It is very easy. You have to just remember the examples, the ratios, and how the expression takes place. So this is deviation from Mendelism, monohybrid cross. Instead of 3:1, phenotypic ratio obtained is 1:2:1. Now let us start with a very interesting concept, multiple allelism. In multiple allelism, when more than 2 alternative forms of a gene in a population occupy the same locus on the chromosome or its homologue. That is, more than 2 alleles for a phenotype or character. Multiple allele. Multiple means what? Many. Many alleles are there. For how many character? 1 character. This is multiple allelism. Multiple alleles may arise due to mutations of a gene. Why multiple allelism? Why there are so many alleles for 1 character? Maybe there is mutation of a gene in the wild type, or different gene may show dominant recessive relations also in multiple allelism. Wonderful example we will be studying here. Sometimes they may show dominant recessive relation, sometimes they may show codominance relation, and usually the wild type is dominant over mutant type. And here crossing over will not take place. Here we'll study about blood group, multiple allelism for ABO blood group. And a picture you can see, it is Dr. Karl Landsteiner, the great man who proposed the ABO blood group system. Now, there are 3 alleles. See, again I repeat, what is multiple allelism? Many alleles for 1 character. What are the character here we are studying? Blood group. And now we are going to study how multiple alleles occur in ABO blood group system. Here the alleles are also called as isoagglutinogenes. Please remember this word. Isoagglutinogenes are alleles for blood group, and they are of 3 types. IA, which is going to represent A blood group, how I will explain. IB and IO. IO is also written as small I because it is the recessive form. Okay, so these are the 3 isoagglutinogenes. Now, Karl Landsteiner, he has described the ABO blood group system in 1901, and afterwards, like late after 31 years, his student Wiener, he proposed the RH system. Anyways, you are going to study about it in some other chapter. Here we will study how blood group is determining by different alleles. So there are different genotypes and the phenotypes. Now, if the genotype, listen carefully students, if it is homozygous IA or heterozygous IA small I, then the blood group is A blood group. If the genotype is IB or IB small I, that is homozygous IB or heterozygous IB, blood group is B. If it is IAIB, they are codominant, both will exist together, so blood group is AB. And if it is a homozygous recessive, either it can be written as like this or it can be written as I0, then the blood group is O blood group. So this you should know. So many different types of alleles are there, and they are responsible for forming any one type of blood group. So multiple allelism. How it is seen? I am going to try to explain you this. Let us draw. Blood group is explained by, now suppose this is the RBC. When Landsteiner carried out experiments, he saw that when blood was transferred from one patient to another, sometimes the patient survived, the recipient survived, but sometimes he died. He studied many people and he saw that in first case, when the RBCs of the person were studied, he found that antigen A was present on the blood group, antigen A. So he called that this blood group of the person will be A, because what antigen is present on the RBC on the plasma membrane? A. He found that in the second person there was another antigen, and this second antigen present in another person was B antigen, and so he called this blood group as B blood group. In the third case, he found that a person had both the antigens, A antigen, B antigen on the plasma membrane. Both the antigens were present, A and B, so he called the blood group as AB. And in the fourth case, he found that there was no antigen present, and so he called it as 0. Now we call it as O blood group because antigens are lacking. He further studied that in the plasma of the first person, in the plasma of the blood, if antigen A is present on the plasma membrane, antibody B was present in the plasma. Second person, antigen B is present in the plasma, antibody A was present. Antibodies I am representing by smaller alphabets. In the third person, antigen AB are present. There are no antibodies present in the plasma of this person. And in the fourth case, when antigen were not there, therefore we call it as zero blood group, both the antibodies are present. So students, you might have known that if antigen A is there, A blood group, antibody present is B. Antigen B, antibody A, blood group B. Both the antigen AB, no antibody, blood group is AB. No antigen, both the antibody, blood group is O. Correct? How is blood group A formed? If you have gene in homozygous condition IA or heterozygous condition IA small I. If you have IB or heterozygous condition IB small I, your blood group will be B. If you are having IA, IB both co-dominant, then your blood group will be AB. And if you are having I0, which can also be represented by recessive homozygous small I, your blood group will be O. So I hope this is clear. Look at this chart, beautiful chart. Blood group A, antigen A. Blood group B, antigen B. Green color it is shown. Blood group AB, both the antigen red and green are there. Blood group O, no antigen. Antibodies, here they have represented by capital, but usually antibodies are represented by small alphabet. In blood group A, antibodies are B. In blood group B, antibodies are A. In AB blood group, no antibodies are there. In O blood group, both the antibodies are there. So I hope you understood. You can think of your blood groups and decide which type of blood group and which type of isoagglutinogens you might be having. So you can see so many alleles are there for one character, and that is blood group. So this is the example of multiple allelism. A person with blood group O can donate blood to all the groups. So he is called as a universal donor. Person with blood group AB can receive the blood from everybody. So he is called as the universal recipient. All these details you will study in circulation chapter. So we need not discuss it here. Here I just wanted to teach you about multiple allelism. Many alleles are responsible for one factor, and that factor is blood group. I hope this is clear. One more example of multiple allelism we'll study in Drosophila, which is responsible for the different wing size. In Drosophila, for the wing size, suppose there is allele Vg+, the Drosophila has normal wings. If the Drosophila has the gene VGNI, it has nicked wings. If it has Vigno, VGNO, then it is having notched wings. You can see in the picture what is notched wing. If VGST, just wings are like strap wings. And if VG is there, only VG, there are no wings or the wings are not at all functional, vestigial wings are present. So one character, what is the character? Shape of the wings. Who is representing? So many alleles are there. So many alleles representing one character, shape of the wing. It is multiple allelism. I hope you understood the example of multiple allelism. We studied multiple allelism in human blood group, ABO blood group system. So many alleles we studied, and multiple allelism in Drosophila, where it is representing the shape of the wings. Now we'll study about pleiotropy. Very interesting. Exact opposite of multiple allelism. In multiple allele, many allele, one character. In pleiotropy, one gene, one allele, one gene, many characters are affected. This is one gene, change in this gene or due to this gene, there is one trait, B trait, 3 trait. So see, it is opposite. Multiple allele, one character, example of multiple alleles. One gene, many characters are affected. It is an example of pleiotropy. In pleiotropy, when a single gene, it is controlling 2 or more different traits, it is called as pleiotropic gene. This phenomenon, it is called as pleiotropy or pleiotropism. According to Mendel, see how it is different from Mendel. What did Mendel say? One gene control only one character. No. According to post-Mendelism, according to pleiotropy, one gene can control many characters. So one gene, sometimes single gene produces 2 related or unrelated phenotypic expressions. For example, sickle cell anemia. It is a disease condition. Let us discuss about sickle cell anemia, how it shows pleiotropy. Before going in detail about sickle cell anemia, let us understand the structure of hemoglobin. This is hemoglobin, which is found in your RBCs. It is made up of 4 chains, 2 alpha chains, 2 beta chains, and these are protein chains, globin chains, and to it is attached the heme, the iron part. Okay, it is sickle cell anemia. It is a autosomal recessive blood disorder which is caused due to mutation on autosome number 11. So there is a gene which is changing on autosome number 11 which is causing sickle cell anemia. It is caused by point mutation. I will explain you what is point mutation. In the beta globulin chain, where is the mutation taking place? Chromosome number 11. Where exactly it is occurring? Because of the change in the chromosome number 11, the beta globulin chain. So there are 2 chains, alpha chains and beta chain. So beta chain is affected. In beta chain, what is the affected? It is affected at the 6th position. Now, globulin is a chain of amino acids. 6th amino acid, it is changed. 6th position, hydrophilic glutamic acid, it is replaced by hydrophobic valine. So instead of glutamic acid, valine is brought. Look at this wonderful diagram. This is a normal one without sickle cell anemia. The DNA had codogen CTC. So RNA produced GAG. When GAG is produced, in the globulin change at the 6th position, glutamic acid is brought and it is a normal hemoglobin and normal function will carried out. But if there is point mutation means only AT becomes ulta, it becomes opposite, it becomes TA. So what is the codogen on the DNA? Instead of CTC, it is CAC. CTC, instead of CTC, it is CAC. What happens on the RNA? Instead of GAG, it becomes GUG. And when it becomes GUG, at the 6th position, instead of glutamic acid, what is brought? Valine is brought. If valine is brought, what happens? Let us see. If normal glutamic acid is produced, these are the normal RBCs, which are oval in shape, biconcave. But because this valine is brought, the shape of the RBC becomes like a sickle vidi. And lesser the space, lesser the concentration of hemoglobin, and then the person, these RBCs, they start clogging and they will block the blood flow. Other symptoms are this person will become anemic because less iron, less oxygen, clogging will take place, hypoxia condition because less oxygen is supplied, frequent infections, immunity is less, swellings of hands and feet will occur, delayed growth or puberty, frequent pain in the chest, abdomen, even in the bones will occur. So this is all occurring in sickle cell anemia. Yes, students. So I hope you enjoyed today's lecture. I enjoyed while teaching. We studied post-Mendelism, neo-Mendelism, how the work which was carried out by different scientists, it differed from the results which were obtained by Mendel. First, we studied about incomplete dominance, where in the flower of Mirabilis jalapa, incomplete or partial expression of the colors were seen. Red color with white gave pink, and the phenotypic ratio changed to 1:2:1. Then we studied codominance. In codominance, we studied cattle coat color. How there also the phenotypic ratio changed because of equal expression. Then we studied wonderful example of multiple allelism, many alleles affecting one character blood group. In human being, many alleles in Drosophila change the shape of the wings. And finally, we studied pleiotropy. One gene, it is bringing about so many different factors, causing so many different symptoms in sickle cell anemia. Next time, I'll show you how this process of sickle cell anemia is inherited. So, thank you. Hello students, this is Mrs. Shweta Ladha from Ferguson College, Pune, your biology teacher. We had started with chapter 3, Inheritance and Variation, and we have finished lots of topics from this chapter. We have discussed with lot of definitions. Then after that we discussed monohybrid cross, dihybrid cross, the Mendel's 3 laws, law of dominant, law of independent assortment, and law of purity of gametes. We also discussed about what is back cross, test cross. After that we studied about post-Mendelism, where we studied about codominance, incomplete dominance, and multiple allelism. If you remember, we had just started with pleiotropism. So what is pleiotropism? Here we are going to study the example of sickle cell anemia. Here there is a single gene which is going to affect many characters. We will study about pleiotropism with the help of sickle cell anemia and its inheritance in detail today. Along with that, I'll be teaching you structure of chromosome, types of chromosome, and distinguishing between X and Y chromosome. And after that, we'll study about few autosomal inheritance pattern in widow speak as well as in ruling tongue. So let us begin with our PowerPoint presentation. So students, let us revise the introduction of pleiotropy. So in pleiotropy, you'll see that there is a single gene, which is affecting many traits. Here in this case, trait A, trait B, and trait C. It is exactly opposite to multiple allelism. In multiple allelism, we had seen it in blood group, human blood group, ABO system. Many alleles affected one trait. Now, when a single gene controls 2 or more different traits, it is called as a pleiotropic gene. Very important. This phenomenon, it is called as pleiotropy, or it is termed as pleiotropism. According to Mendel, one gene controls one character, but here sometimes post-Mendelism it was proved that single gene, it produces 2 related or unrelated phenotypic expressions. For example, in sickle cell anemia. Let us see what happens in sickle cell anemia. Before that, let us understand the structure of hemoglobin. Hemoglobin is a conjugate protein made up of 2 parts. Heme means iron and globin means chain of amino acid, that is protein. And there are 4 chains as you can see in the picture. Alpha chain 1, alpha chain 2, beta chain 1, beta chain 2 along with the heme particle. So 4 hemoglobin chains are there. The globin chains, 2 alpha chains, 2 beta chains are there in the hemoglobin along with iron. Sickle cell anemia, it is an autosomal recessive blood disorder caused due to mutation on autosome, chromosome number 11. Autosomal, I will again explain you in the next slide what is an autosome, which is responsible for somatic characters. This sickle cell anemia is caused due to mutation on chromosome number 11 and it is a recessive disorder. Recessive. Be shown only if it is in homozygous condition. It is caused due to point mutation, very important term. Where is the point mutation occurring? What is the result? It occurs in beta globulin chain, in this chain of the hemoglobin. At the 6th position, hydrophilic glutamic acid, it is replaced by hydrophobic valine amino acid. Let us see this in detail. So if you see in a normal DNA, for normal formation of hemoglobin, the codogen present on the DNA is CTC. Because of that, mRNA has codon GAG, and when GAG is there, glutamic acid, which is a hydrophilic amino acid, is there at 6th position. But because of point mutation, what is point mutation? You can see here, instead of AT, this one base pair, nucleotide base pair is altered, it becomes TA. So the codogen, instead of CTC, it is becoming CAC. Listen carefully. What type of mutation is occurring? Point mutation. Which chromosome? Chromosome number 11. What happens? The codogen changes from CTC to CAC. And so what happens on the RNA? The codon becomes GUG instead of GAG. This change in one place at the nucleotide brings different amino acid. Instead of glutamic acid, now valine is brought. Now if valine is placed at the 6th position, the entire structure of hemoglobin changes. Let us see what happens. The hemoglobin, the globin chain changes, and that's why the shape of the RBC changes. Normally the RBC are oval in shape, biconcave, isn't it? Enucleated. But because of the change at the 6th position in the globin, beta globulin chain, the shape of the RBC become like a sickle. Marathi मध्ये विळी म्हणतो आपण. It becomes like a sickle. Now, as it is becoming like a sickle, it is not able to flow properly in the blood vessels and it starts clogging the blood vessels. So, you can see this is a normal hemoglobin which is placed in the RBC biconcave disc, and here the abnormal hemoglobin, because of abnormal shape, sickle shape hemoglobin, it is not able to flow properly in the blood vessels. The other symptoms, other traits because of this are the person who is having sickle cell anemia shows anemic condition, anemia because less iron, because less shape, the shape of the hemoglobin is not proper, less iron, so anemic condition. I showed you how blood clogging occurs, plaques are formed, blood flow is obstructed, less supply of oxygen is given to the organs, so hypoxia. Hypo means less. Oxia means supply of oxygen. Less supply of oxygen is done. This person is supposed to get frequent infections. Swellings of hands and feet takes place. There is delayed growth or puberty. And there is frequent pain in the chest, abdomen and in the bones. So see, the change, the mutation at one gene is responsible for so many traits. Thus, it is an example of pleiotropy. Now let us see how it is inherited, what are the genotypes and what are the phenotypes. If suppose it is the genotype is HbA, which is homozygous dominant, then the person will have normal hemoglobin. Hb stands for hemoglobin, and this genotype is, if it is homozygous dominant, normal hemoglobin will be formed. The person will not have sickle cell anemia and he will lead a normal life. But if it is, the person is having HbA, HbS in heterozygous condition, this person is believed to be a carrier for sickle cell anemia. This person will have little or mild anemic condition because their cells become sickle cell trait. Of course, but this person does not die. But as this is a recessive gene, if the genotype of the person is homozygous recessive, HbS, HbS, then 100% the person will develop sickle cell anemia, the shape of the hemoglobin will change, hypoxic condition, due to anemic condition, the child will die. So it is a lethal gene. Okay, now let us see its inheritance pattern. Now suppose two person, we are considering parents, they are sickle cell carriers. If they are carriers, they are heterozygotes, isn't it? So HbS, HbA, HbS, HbA. They are marrying with each other. What will be the gametes produced? Here the gametes will be HbS, HbA. Here the gametes produced will be HbS, HbA. The offsprings produced in F1 generation, if you see, HbS, HbS, homozygous recessive. Obviously, what will happen to this child? He is suffering from sickle cell and he dies because it is a lethal gene. If the child gets HbA and HbA, it is a case of homozygous, it should be HbS, HbA. Heterozygous condition is there, and this heterozygous condition, these people, they are sickle cell carriers. And how many are they? 1 and 2. So this 2. And one case we'll see that if HbA comes with HbA, it is a homozygous normal. He doesn't have sickle cell trait, and this is 1. So see how it deviated from the normal Mendelian ratio. According to Mendel, the normal ratio should be 3:1, but here it is 2:1. And here one gene is controlling many traits, pleiotropy. So this is neo-Mendelism, going away from the conclusions which Mendel gave. I hope you followed. So it is a marriage between two sickle cell carriers. It will produce one normal. This was the normal one having homozygous dominant HbA, HbA. 2 are carriers which are heterozygous, HbS, HbA, and one will be having sickle cell anemia, lethal gene, and this child will die, which is having homozygous recessive gene HbS, HbS. So I hope you understood the concept of pleiotropy and how it is one gene is affecting more than one trait. Sickle cell anemia. A long question can be asked for 3 marks on this part, or an MCQ can be asked on the phenotype, genotype. So prepare this, practice this chart again and again. Now let us proceed. Now I am going to start with structure of chromosomes. Now chromosomes, chromo colored, soma body. They are called as colored bodies because they get stained with dyes. These chromosomes are thread-like structures. They are made up of nucleic acids. They are having proteins and they are found in nucleus of most of the eukaryotic cells. So these colored bodies, thread-like structures made up of nucleic acid and protein found in nucleus of eukaryotic cell. Eukaryotic having a well-developed, well-defined nucleus. What do they carry? They carry genetic information in the form of genes. The term was coined by W. Waldeyer. Very important. Learn it by heart again and again. Can be asked for MCQ. So the term chromosome was coined by W. Waldeyer in 1888. Size varies from species to species. It can be from 0.1 to 0.33 micrometer in length to 0.2 to 2 micrometer in thickness. So the size of the chromosome, the length of the chromosome is different, and even in the number of chromosome is different from species to species. Chromosomes in human beings can be divided into two types. We have autosomes, which are also called as the body chromosomes, and we have the allosomes, which are also called as the sex chromosome. This is again important, students. Our chromosomes are categorized into two groups, autosomes and allosomes. We have 22 pairs of autosomes, that is 44, and we have one pair of allosome, that is sex chromosome. And of course, you know the difference of allosomes between a male and a female. So in human, each cell, any cell of your body, it contains 23 pairs of chromosomes. 23 pairs means how many in total? 46 chromosomes. 22 of these pairs are autosomes, responsible for somatic characters, body characters. They look the same in both and females. They are similar. These 22 pairs are similar in males and females, and usually we write them as like this: 22 pairs of autosomes. They are similar in males and females. The 23rd pair, the sex chromosome, the allosome, it is different in male and female. In males, it is XY, and in females, it is XX. Now, let us see the typical structure of a chromosome. You can go through this picture. I have taken this picture from your textbook itself. So, we'll begin. Chromosome consists of two identical, similar, spirally coiled, which are coiled threads, which are called as sister chromatids. You can look at this picture. This is a chromatid. This is a chromatid. If you look under the microscope, you will see that they are made up of spirally coiled structures. And these spirally coiled structures, thread-like structures, are called as sister chromatid. So this and this, they are sister chromatids of each other. They are joined at the centromere. This region is called as the centromere. It is also called as primary constriction. So they are joined at the centromere like this. Okay? Each centromere, it is having a disc-shaped structure. On this, I'll show it with some other color. We'll show it with green color. There is a disc-like structure on the centromere, and this disc-like structure present on the centromere, it is called as kinetochore. What is the function of kinetochore? It provides space for attachment of spindle fibers during cell division. You have studied mitosis, meiosis in 11th standard. You know that how chromosomes are pulled apart during anaphase. And the spindle fibers, they attach to these kinetochores and pull the sister chromatids apart during cell division. Depending on number of centromere, chromosomes can be monocentric, dicentric or polycentric. If you look at the second picture, you will see there is here one primary constriction, which is centromere 1, which will have this kinetochore, okay, and where the sister chromatids are attached. You can see there is one more secondary constriction at the bottom and one more secondary constriction. So there are more constrictions. When there are more constriction, it will be when there is single monocentric, two centromeres dicentric, many centromeres polycentric. So I hope you understood. Now in this chromosome, the secondary constriction, above it there is a region which is called as satellite. Okay, this is the satellite. This secondary constriction is also called as nucleolar organizer because it helps or takes plays important role in synthesis of nucleolus. I hope you know what is a nucleus. This is a cell. This is nucleus. This is nucleolus. And these are the chromosomes. So the satellite chromosomes, just near the sat chromosomes, there is a secondary constriction. It is associated with synthesis of nucleolus. Important function. You should know. What is the function of kinetochore? Providing space for attachment of spindle fibers. What is the function of secondary constriction, which is also called as nucleolar organizer? It plays important role in synthesis of nucleolus. Let us proceed. Each chromatid is made of 2 to 4 or 8 thread-like structure which are called as chromonymata. Further, the chromosome is made up of, so each chromatid, this was one chromatid, this one, second, three. This is internally again made up of coiled thread-like structures which are called as chromonymata. What is the chromonymata made up of? We know the nucleosome structure. It is made up of DNA and a protein coat, histone proteins. It is darkly stained network of chromatin fibers having two regions. This picture is very nice. You can see here, there are two types of regions. One region is darkly stained, other is lightly stained. Okay? The genetically active euchromatin and the condensed transcriptionally inactive heterochromatin is present. You can see in this picture, this is euchromatin. What is euchromatin? It is little bit lightly stained. It is genetically active. It will participate in protein synthesis. Whereas the heterochromatin, the other region, you can see this which is stained with orange color. It is inactive part. Okay. So I hope you understood the two regions of chromosome, euchromatin, heterochromatin. Euchromatin is active region. Heterochromatin is inactive in transcription. It has some other functions, other roles. Usually, chromosomes are stained by basic Fulgen stains because chromosome, it is a nucleic acid. It is made up of acid. So, Fulgen stain, basic stains are used. I told you about SAT chromosome having satellite, having secondary constriction which plays important role in synthesis of nucleolus, which is also called as nucleolar organizing region. How many chromosomes are having this nucleolar organizing region? Only chromosome number 13, 14, 15, 21, 22. Only autosome number 13, 14, 15, 21 and 22 are SAT chromosomes having nucleolar organizer region which will play important role in synthesizing the nucleolus. All autosomes or all chromosomes are not having the SAT chromosome. I hope you are getting these points clear. Are chromosomes visible all the time? No. Chromosomes are visible only during cell division. I hope you remember the cell cycle you studied last year. Cell cycle, it consists of M phase and then it consists of a very big interphase, and this interphase is then divided into G1 growth phase, synthetic phase, S phase and G2 phase. During this interphase, chromosomes are not visible. Chromosomes are visible only during M phase. In M phase also, in prophase, they are not visible. They are a form of chromatin fibers. They will be visible after prophase, that is metaphase, anaphase, telophase. And you might have performed an experiment last year where you studied the onion root tip cells, where you studied mitotic divisions and you have seen the chromosomes. Always remember, students, chromosomes are visible only during cell division. That too which phases? Metaphase, anaphase and telophase. Especially very clearly visible in metaphase and anaphase. Otherwise, in prophase, they are in the form of chromatin network, and even here in interphase, they are not visible. They are capable of self-replication. They can self-replicate and they play a very important role in heredity. You know, you are like your parents. Somebody might be like mother, somebody like maybe like father, somebody might be like grandmother or grandparents, anybody. This is because of our genes which we get through our chromosomes. They play important role in mutation. Sometimes our gene mutate, and where are these genes located? They are located on chromosome. They bring about variation, and if variation occurs, it will lead to evolution. So I hope you are getting this. One more thing I would like to tell, if you see this structure of chromosome, this upper part, it is sometimes called as P arm, which is present above the centromere, and the lower is called as the Q arm. These two chromosomes, these two branches, or we can say these two chromatids which are present in the same chromosome are sister chromatids, but this chromosome, suppose I call it A, this chromosome, suppose I call it B. A and B, they are homologous chromosomes. It is a homologous pair, and the branches or the chromatids of the homologous pair, they are called as non-sister chromatids. You should be clear about this too. These two are sister chromatids, but these two are non-sister chromatids. Centromere, primary constriction, kinetic core, we have already discussed. Euchromatin, active region, heterochromatin, inactive for transcription. Of course, it has some other functions. So this is all about the structure. Now let us see the types of chromosomes on basis of position of centromere. Very important, can be asked for MCQ, one-line question, diagram for 2 marks, match the pair. So listen carefully, and it is very simple. You know now what is centromere. Centromere is that primary constriction where the chromatids are attached. Now the position of this centromere, it varies. So there are 4 types of chromosomes. First is telocentric. Look at this picture. Telocentric. The centromere is at the tip. It is at the tip, and so only one arm is seen. It is I-shaped chromosome. Listen students carefully. I-shaped chromosome is telocentric. Centromere is at the tip, at the end. Only one arm is there. Acrocentric. The centromere is towards the end. Very important. Near the end, not at the end. Therefore, it appears J-shaped. And such type of chromosome, it is called as acrocentric chromosome. The next type is submetacentric. The centromere is towards the center. These words are important because in MCQ they will be using these words only. When it is towards the center, it appears L-shaped. Look at this picture, L. Okay, one arm is short, one arm is long, and this is called as submetacentric. Whereas the 4th type is metacentric. The centromere is at exact center. The 2 arms are equal and it appears V-shaped. Thus, this is metacentric. So, the 4 types of chromosomes depending on the basis of position of centromere are telocentric, I-shaped, centromere at the end, acrocentric, J-shaped, centromere towards the end, submetacentric, L-shaped, centromere towards the center, metacentric, V-shaped, centromere at the center. Diagrams can be asked, match the pairs, MCQs, one line. So please draw this and practice it. Now further we'll go and study difference between X and Y chromosome. X chromosome, we know that females have XX sex chromosome, males have XY chromosome. So what is the difference between X and Y chromosome? X chromosome is comparatively longer. So suppose this is a centromere and it is comparatively longer. Y chromosome, I will draw it here, it is comparatively shorter. First difference. Second, X chromosome, it is metacentric. See, V-shaped. Both the arms are equal and the centromere is at the center, whereas Y chromosome is acrocentric. Can be asked in MCQ. Remember carefully. X chromosome has larger amount of euchromatin. So it is more active, genetically more active, will participate in more transcription, more protein formation. Whereas Y chromosome has lesser amount of euchromatin. So it is genetically less active. X chromosome carries sex-linked genes on the non-homologous part of sex-linked genes. Let us see. This upper part of the X chromosome and upper part of the Y chromosome, this is called as the non-homologous part because the genes vary in this region. Whereas I'll draw it with other color. Let us see, this part is homologous part. So this part is homologous part. Listen carefully. The genes that are located on the non-homologous part of X chromosome here, they are called as sex-linked genes. Okay, and the genes that are present on the non-homologous part of the Y chromosomes are called as Hollandric genes. These are Hollandric genes. Usually, sex-linked genes, they show crisscross inheritance. Crisscross, in one generation it will be with the mother, then it is given to the son, then to son's daughter, so we'll call it as granddaughter. So crisscross inheritance, but Hollandric genes, they show straight inheritance. Father will give the genes to the son because only males have Y chromosomes, but X chromosomes are both present in both males and females. So students, 2 marks question can be asked on this topic. Differentiate between X and Y chromosome. Look at this picture again. I have taken it from your textbook. So this is X chromosome, this is Y chromosome. This is metacentric, acrocentric. Genetically more active, less active. Upper region, non-homologous part. Here it is carrying sex-linked genes. Here it is carrying Hollandric genes. So I'll write just H, Hollandric genes, sex-linked genes. Hollandric gene shows straight inheritance, father to son. Sex-linked gene shows crisscross inheritance, mother to son, from son to granddaughter, again granddaughter to her son, great-great-grandson. So crisscross inheritance. And this lower region, it is homologous, similar type of genes are present. So I hope you understood. Difference between X and Y chromosome diagram can also be asked. Let us now proceed with autosomal inheritance. You know now what are autosomes? The chromosomes which are responsible for body characters are autosomes. They also control a variety of traits but not sex trait, body characters. These traits are called as autosomal traits and their inheritance is called as autosomal inheritance. Some characters are influenced by dominant genes while others are recessive. Let us see autosomal dominant in inheritance. 2 examples, widow's peak and rolling tongue. Now we are going to discuss only about widow's peak. Autosomal recessive inheritance include phenylketonuria, cystic fibrosis and sickle cell anemia. I have already taught you about sickle cell anemia. Let us see about widow's peak. It is a V-shaped hairline present on the forehead. It is determined by autosomal dominant gene. It is just a character, students. It is not a disease. Widow's peak occurs in homozygous condition. If you are having capital W condition or heterozygous capital W, small w condition, then you will have widow's peak. But if you are having a recessive homozygous gene, small w, you will have a straight hairline pattern. Both males and females have equal chances of inheritance. So you can see this lady, this person is having widow's peak, so it might be having homozygous or heterozygous condition. Here it is having a straight hairline. It is not a disease. It is just a routine character. So these characters, they are inherited by autosomes. They are dominant genes, so it is autosomal dominant inheritance. There is one more example, rolling tongue, which I'll teach you in next lecture. So I hope you understood widow's peak. Yes, students. So now today we discuss 3 important concepts. Pleiotropy. In pleiotropy, in detail, I taught you about sickle cell anemia, its inheritance. Then we studied the structure of a chromosome, typical chromosome. We studied about SAT chromosomes. We studied about 4 types of chromosomes depending upon position of centromere. We distinguished between X and Y chromosome. And finally, we started with autosomal inheritance where we studied about widow's peak. I hope you have understood all the points. Please revise it once again. Thank you.
केंद्र सरकारनं शेतकऱ्यांच्या हितासाठी कृषी कायद्यात केलेल्या सुधारणा भारतीय कृषी क्षेत्रात एका नवीन अध्यायाची सुरुवात ठरतील केंद्रीय कृषी मंत्र्यांचं शेतकऱ्यांना पत्राद्वारे आश्वासन. शेतकरी बंधू भगिनींशी खुल्या मनानं संवाद साधणारं हे पत्र शेतकऱ्यांनी आणि देशवासियांनी अवश्य वाचावं, पंतप्रधान नरेंद्र मोदी यांचं आवाहन. शेतकऱ्यांना आपल्या हक्कांसाठी आंदोलन करण्याचा अधिकार असला तरी यामुळे इतरांचं जीवन विस्कळीत होणार नाही याची खबरदारी घ्यावी, सर्वोच्च न्यायालयाची स्पष्टोक्ती. लक्षावधी देशवासियांचं भरणपोषण करणारा शेती हा एकमेव व्यवसाय कोरोना काळात अर्थव्यवस्थेला आधार देत होता, उपराष्ट्रपतींकडून कृषी क्षेत्राचं कौतुक. देशांतर्गत उद्योग क्षेत्रातून 27,000 कोटी रुपयांची संरक्षण सामग्रीची खरेदी करायला केंद्र सरकारची परवानगी. राज्य सरकारी अधिकारी आणि कर्मचाऱ्यांसाठी असणाऱ्या वैद्यकीय खर्च प्रतिपूर्तीमध्ये कोरोना आजाराचा समावेश करण्याचा राज्य सरकारचा निर्णय. देशाचं पहिलं आंतरराष्ट्रीय क्रीडा विद्यापीठ महाराष्ट्रात येत्या शैक्षणिक वर्षापासून प्रवेश प्रक्रिया सुरू. देशात उपचाराधीन रुग्णांचं प्रमाण कमी होऊन एकूण रुग्णसंख्येच्या 3.14% आणि बरे होण्याचं प्रमाण वाढून 95.40%. आणि ॲडलेड इथल्या पहिल्या क्रिकेट कसोटी सामन्यात आज दुसऱ्या दिवशी भारतीय संघाचा पहिला डाव 244 धावात संपुष्टात. नमस्कार. 11च्या बातमीपत्रात मी जाई वैशंपायन आपलं स्वागत करते. पाहूया बातम्या विस्तारानं. केंद्र सरकारनं शेतकऱ्यांच्या हितासाठी कृषी कायद्यात केलेल्या सुधारणा भारतीय कृषी क्षेत्रात एका नवीन अध्यायाची सुरुवात ठरतील असा विश्वास केंद्रीय कृषी मंत्री नरेंद्रसिंग तोमर यांनी देशातल्या शेतकऱ्यांना लिहिलेल्या पत्राद्वारे व्यक्त केला आहे. हे सुधारित कायदे शेतकऱ्यांना अधिक स्वावलंबी आणि सक्षम करतील असं कृषी मंत्र्यांनी म्हटलं आहे. गेल्या काही दिवसांमध्ये अनेक शेतकरी संघटनांनी कृषी कायद्यांना पाठिंबा दर्शवला आहे असं तोमर यांनी म्हटलं आहे. नवीन सुधारणांनुसार कृषी मालासाठी MSP अर्थात किमान आधारभूत किंमत धोरण यापुढेही कायम राहील अशी ग्वाही त्यांनी दिली आहे. केंद्र सरकार शेतकऱ्यांकडून किमान आधारभूत किमतीवर आधारित कृषी मालाची खरेदी मोठ्या प्रमाणावर करत आहे. त्यासाठी अशा खरेदी केंद्रांची संख्याही वाढवली आहे असं सांगून समाजातले काही घटक मात्र याबाबत गैरसमज पसरवत आहेत असा आरोप त्यांनी केला. कृषी उत्पन्न बाजार समित्यांचे बळकटीकरण करण्यात येत असून या समित्या वगळता खासगी बाजारपेठांमधल्या व्यवहारांवर कर आकारायला राज्य सरकारांना मुभा असेल असं कृषी मंत्र्यांनी स्पष्ट केलं. कृषी संबंधित वादांसाठी न्यायालयात न्याय मागण्याचा अधिकार शेतकऱ्यांना असेल तर कृषी कराराची नोंदणी करण्याचा अधिकार राज्य सरकारांना प्रदान केला जाईल. शेतकऱ्यांच्या जमिनी कोणत्याही परिस्थितीत बळकावल्या जाणार नाहीत. जमिनीचं हस्तांतरण, विक्री, भाडेतत्व किंवा जमीन गहाण ठेवण्याचा अधिकार कोणालाही दिला जाणार नाही. कंत्राटदाराला शेतकऱ्यांच्या जमिनीवर कोणतेही कायमस्वरूपी बदल करता येणार नाहीत. तसंच कंत्राटदारांनी केलेल्या तात्पुरत्या बदलांच्या आधारावर कंत्राटदाराला कर्ज मिळणार नाही असं नरेंद्रसिंग तोमर यांनी या पत्राद्वारे स्पष्ट केलं आहे. केंद्र सरकार सबका साथ, सबका विकास, सबका विश्वास या मंत्रानुसार मार्गक्रमण करत असून कोणताही भेदभाव न करता देशातल्या प्रत्येक नागरिकाचं हित जपायचा प्रयत्न करत आहे असं त्यांनी म्हटलं आहे. कृषी मंत्र्यांच्या या पत्राबद्दल पंतप्रधान नरेंद्र मोदी यांनी संतोष व्यक्त केला आहे. शेतकरी बंधू भगिनींशी खुल्या मनानं संवाद साधून कृषी मंत्र्यांनी या संदर्भातले गैरसमज दूर केले आहेत. हे पत्र शेतकऱ्यांनी अवश्य वाचावं तसंच ते जास्तीत जास्त नागरिकांनीही वाचावं या दृष्टीनं प्रयत्न झाले पाहिजेत असं पंतप्रधानांनी ट्विटर संदेशात म्हटलं आहे. शेतकऱ्यांना आपल्या हक्कांसाठी आंदोलन करण्याचा अधिकार असला तरी त्यामुळे इतरांचं जीवन विस्कळीत होणार नाही याची खबरदारी घेतली पाहिजे असं सर्वोच्च न्यायालयानं स्पष्ट केलं आहे. कृषी कायद्यांच्या विरोधात सुरू असलेल्या शेतकरी आंदोलनाच्या विरोधात दाखल याचिकेवर सुनावणी करताना सर्वोच्च न्यायालयानं यावर टिप्पणी केली. आंदोलनाच्या काळात मालमत्ता किंवा जीवितहानी होणार नाही याकडे शेतकरी संघटनांनी लक्ष द्यावं असे निर्देश न्यायालयानं दिले. केवळ आंदोलन करून कोणताही प्रश्न सुटणार नाही. त्यासाठी केंद्र सरकारबरोबर सातत्यपूर्ण चर्चा होणं आवश्यक आहे. कदाचित या चर्चेच्या पहिल्या फेऱ्यांमध्ये सकारात्मक निष्कर्ष निघू शकणार नाही, मात्र तरीही चर्चा सुरू ठेवली पाहिजे असं न्यायालयानं सांगितलं आहे. याकरता केंद्र सरकार आणि शेतकरी संघटनांच्या प्रतिनिधींची समिती स्थापन करावी असा सल्ला न्यायालयानं दिला आहे. या समितीद्वारे सुवर्णमध्य साधण्याचा प्रयत्न व्हावा तसंच जोपर्यंत सर्वमान्य तोडगा निघत नाही तोपर्यंत शांततामय मार्गानं आंदोलन सुरू ठेवायला हरकत नाही असं न्यायालयानं म्हटलं आहे. लक्षावधी देशवासियांचं भरणपोषण करणारा शेती हा एकमेव व्यवसाय कोरोना काळात जोमानं सुरू होता आणि अर्थव्यवस्थेला आधार देत होता, अशा शब्दात उपराष्ट्रपती एम व्यंकय्या नायडू यांनी कृषी क्षेत्राचं कौतुक केलं आहे. ते काल कोइंबतूर इथल्या तमिळनाडू कृषी विद्यापीठाच्या पदवीदान समारंभात बोलत होते. भारताच्या अर्थव्यवस्थेचा आणि संस्कृतीचाही अविभाज्य घटक असणाऱ्या शेतीला हवामान बदलाचा सामना करण्यासाठी सक्षम आणि अधिक उत्पादनक्षम करणं ही काळाची गरज आहे, असंही ते म्हणाले. केंद्र सरकारनं बियाणं, कीटकनाशकं, खतं आणि पतपुरवठ्याची वेळेवर व्यवस्था केल्यामुळे कोरोना काळात खरीपाखालचं क्षेत्र गेल्या वर्षीच्या तुलनेत 59 लाख हेक्टरनी वाढल्याची माहिती त्यांनी दिली. येत्या काळात अल्पभूधारक शेतकऱ्यांच्या हिताच्या दृष्टीनं तंत्रज्ञान आणि शेतीची तंत्र विकसित करावीत, असं आवाहन उपराष्ट्रपतींनी यावेळी नव्या पदवीधरांना केलं. पंतप्रधान नरेंद्र मोदी आज मध्य प्रदेशातल्या शेतकऱ्यांशी दूरदृश्य प्रणालीद्वारे संवाद साधणार आहेत. मध्य प्रदेश सरकारनं आयोजित केलेल्या पंतप्रधान किसान कल्याण कार्यक्रमात शेतकऱ्यांच्या थेट बँक खात्यात सुमारे 1600 कोटी रुपयांचा निधी यावेळी हस्तांतरित केला जाणार आहे. याशिवाय शेतकऱ्यांना किसान क्रेडिट कार्डांचं वाटपही केलं जाणार आहे. सुधारित कृषी कायद्यांबाबत शेतकऱ्यांना माहिती दिली जाणार आहे. मध्य प्रदेशचे मुख्यमंत्री शिवराज सिंग चौहान या कार्यक्रमाला उपस्थित असतील. याशिवाय प्रत्येक जिल्ह्यात अशा प्रकारचं संमेलन आयोजित करण्यात येणार आहे. चालू खरीप विपणन हंगामात केंद्र सरकारनं देशभरातल्या शेतकऱ्यांकडून 398 लाख टन धान खरेदी केलं आहे. या अंतर्गत सुमारे 75,263 कोटी रुपयांची खरेदी झाली असून त्यामुळे 45,92,000 शेतकऱ्यांना लाभ झाला आहे. महाराष्ट्रासह पंजाब, हरियाणा, उत्तर प्रदेश, तेलंगण, उत्तराखंड आणि इतर राज्यातल्या शेतकऱ्यांकडून धान खरेदी केल्याची माहिती कृषी मंत्रालयानं दिली आहे. देशांतर्गत उद्योग क्षेत्रातून संरक्षण सामग्रीची 27,000 कोटी रुपयांची खरेदी करायला केंद्र सरकारनं परवानगी दिली. संरक्षण मंत्री राजनाथ सिंग यांच्या अध्यक्षतेखाली काल DAC अर्थात संरक्षण खरेदी परिषदेची बैठक झाली. त्यावेळी हा निर्णय घेण्यात आला. लष्कर, नौदल आणि वायुदलाला लागणाऱ्या विविध शस्त्रास्त्र, प्रणाली आणि अन्य सामग्रीची खरेदी यात समाविष्ट आहे. संरक्षण खरेदी प्रक्रिया 2020 अंतर्गत झालेली या परिषदेची ही पहिलीच बैठक होती. देशांतर्गत उद्योगातून खरेदी करण्याच्या निर्णयामुळे मेक इन इंडिया आणि आत्मनिर्भर भारत अभियानाला गती मिळू शकणार आहे. वायुदलासाठी हवेत काम करणारी पूर्वसूचना आणि नियंत्रण प्रणाली, नौदलासाठी गस्ती जहाजं आणि लष्करासाठी मॉड्युलर पुलांच्या खरेदीचे प्रस्ताव काल मंजूर करण्यात आले. वंदे भारत अभियानाच्या 1 डिसेंबरपासून सुरू झालेल्या फेज 8+ या टप्प्याअंतर्गत सुमारे 1,30,000 व्यक्तींना मायदेशी परत आणण्यात आलं आहे. आतापर्यंत एकूण 38,40,000 व्यक्तींना परत आणण्यात आल्याची माहिती परराष्ट्र मंत्रालयाचे प्रवक्ते अनुराग श्रीवास्तव यांनी काल नवी दिल्लीत पत्रकार परिषदेत दिली. कोरोना संकट काळात परदेशी अडकलेल्या भारतीयांना सुखरूप आणण्यासाठी मे महिन्यात वंदे भारत मिशनची सुरुवात करण्यात आली होती. सध्या चीनमधल्या 2 बंदरांमध्ये 2 मालवाहतूक जहाजांवर 39 भारतीय नागरिक अडकले असून त्यांना भारतात आणण्यासाठी प्रयत्न सुरू आहेत. कोविड-19 संबंधीच्या निर्बंधांमुळे या जहाजांमधला माल बंदरांवर उतरवणं सध्या शक्य नाही. त्यामुळे चीन सरकारकडून या नागरिकांना परत आणण्यासाठी परवानगीची वाट पाहत असल्याचं श्रीवास्तव यांनी स्पष्ट केलं. आरोग्य मंत्रालयानं आज दिलेल्या माहितीनुसार देशात काल 22,889 नव्या कोरोनाबाधित रुग्णांची नोंद झाली. 338 रुग्णांचा कोरोनामुळे मृत्यू झाला तर 31,087 रुग्ण कोरोनामुक्त होऊन घरी परतले. उपचाराधीन रुग्णांची संख्या आणखी कमी होऊन आता 3,13,831 इतकी झाली आहे. हे प्रमाण एकूण रुग्णसंख्येच्या 3.14% उतरलं आहे. आतापर्यंत देशभरातले एकूण 95,20,827 रुग्ण कोरोनामुक्त झाले असून कोरोना रुग्ण बरे होण्याचं प्रमाण वाढून 95.40% पोहोचलं आहे. देशातली कोरोनामुळे दगावलेल्यांची एकूण संख्या 1,44,789 वर पोहोचली आहे. सध्या देशाचा कोरोना मृत्यू दर 1.45% स्थिर आहे. भारतीय स्वदेशी बनावटीच्या कोव्हॅक्सिन या लसीमुळे रोगप्रतिकार शक्तीत मोठ्या प्रमाणात वाढ होत असल्याचं क्लिनिकल चाचण्यांमधून सिद्ध झालं आहे. या लसीची निर्मिती हैदराबादची भारत बायोटेक आणि ICMR एकत्रितपणे करत आहेत. कोव्हॅक्सिन लसीची चाचणी होत असलेल्या स्वयंसेवकांवर या लसीचे कोणतेही दुष्परिणाम होत नसून ही लस वापरासाठी सुरक्षित असल्याचं चाचण्यांच्या पहिल्या टप्प्यात सिद्ध झालं आहे. कोवॅक्सिनची साठवणूक 2 ते 8 अंश सेल्सिअस तापमानात होऊ शकत असल्यामुळे ती राष्ट्रीय लसीकरण योजनेच्या मापदंडांमध्ये बसणारी लस असल्याचं ICMRनं म्हटलं आहे. सध्या या लसीच्या क्लिनिकल चाचण्यांचा तिसरा आणि शेवटचा टप्पा सुरू असून यात 18 ते 55 वर्ष वयोगटाच्या 26,000 स्वयंसेवकांवर चाचणी घेतली जात आहे. जगभरात आतापर्यंत कोरोना रुग्णांची संख्या 7,52,00,000पेक्षा जास्त झाली आहे आणि आतापर्यंत 16,67,000पेक्षा जास्त लोकांचा यामुळे मृत्यू झाला आहे. कोरोना संसर्गामुळे अमेरिका सर्वाधिक बाधित देश असून तिथं कोरोना बाधितांची संख्या 1,76,00,000पेक्षा जास्त झाली आहे. या आजारामुळे अमेरिकेत 3,17,000पेक्षा जास्त व्यक्तींचा मृत्यू झाला आहे. ब्राझीलमध्ये 71,11,000पेक्षा जास्त जण कोरोना बाधित झाले आहेत. राज्यात काल 4,358 कोरोनाबाधित रुग्ण बरे होऊन घरी परतले. 3,880 नव्या रुग्णांची नोंद झाली तर 65 जणांचा कोरोनामुळे मृत्यू झाला. राज्यातलं कोरोना रुग्ण बरे होण्याचं प्रमाण वाढून 94.14% इतकं झालं आहे. आतापर्यंत राज्यभरातले एकूण 17,74,255 रुग्ण कोरोनामुक्त झाले आहेत. राज्यातली एकूण बाधितांची संख्या 18,84,773 झाली आहे तर एकूण कोरोना बळींची संख्या 48,499 इतकी झाली आहे. राज्याच्या कोरोना मृत्यूदरात घट होऊन तो 2.57% पोहोचला आहे. सध्या राज्यभरात 60,905 कोरोनाबाधितांवर उपचार सुरू आहेत. येत्या 2 वर्षात देशभरात पथकर वसुलीसाठी संपूर्णपणे GPS तंत्रज्ञानाचा वापर करून पथकर नाके बंद करण्याचा निर्णय घेतल्याची माहिती केंद्रीय रस्ते वाहतूक आणि महामार्ग मंत्री नितीन गडकरी यांनी दिली आहे. ते काल नवी दिल्लीत ॲसोचॅमच्या स्थापना सप्ताह कार्यक्रमात बोलत होते. वाहनाच्या हालचालीनुसार पथकराची वसुली थेट बँकेच्या खात्यातून होईल असंही ते म्हणाले. सध्या व्यापारी उपयोगाच्या सर्व वाहनांवर ट्रॅकिंग प्रणाली बसवलेली असतेच. आता जुन्या वाहनांवरही GPS बसवण्यासाठी सरकार काहीतरी कार्यक्रम तयार करेल असं गडकरी म्हणाले. येत्या मार्चपर्यंत 34,000 कोटी इतकी पथकर वसुली होईल अशी आशा त्यांनी व्यक्त केली. GPS तंत्रज्ञान वापरण्यामुळे येत्या 5 वर्षात हा आकडा 1,34,000 कोटीपर्यंत जाईल असा अंदाजही त्यांनी व्यक्त केला. 2020 हे वर्ष लक्षात राहिलं ते कोरोनाच्या अभूतपूर्व संकटासाठी. पण संकटाच्या या काळातही देशात आणि जगभरात अनेक महत्त्वाच्या घडामोडी घडल्या. या घडामोडींचा आढावा घेणार आहोत दृष्टिक्षेप 2020 या कार्यक्रमातून.
जब तक दवाई नहीं तब तक ढिलाई नहीं.
माझे कुटुंब माझी जबाबदारी.
हम भारत को आत्मनिर्भर बनाकर रहेंगे. देश में MSP रहेगी और किसानों को कहीं पर भी अपनी उपज बेचने की आजादी भी रहेगी. भारत पूरी दृढ़ता से देश की एक एक इंच जमीन की, देश के स्वाभिमान की रक्षा करेगा. भारत माता की जय. सियावर रामचंद्र की जय. सिया राम.
CAA पास होने के बाद देश भर में भ्रम फैलाने का काम किया.
या वर्षभरातल्या महत्त्वाच्या घटनांचा आढावा घेणार आहोत दृष्टिक्षेप 2020 या 6 भागांच्या मालिकेतून. तेव्हा पाहायला विसरू नका. 26 डिसेंबर ते 31 डिसेंबर या कालावधीत रोज रात्री 8:30 ते 9:30 या वेळेत दृष्टिक्षेप 2020 फक्त सह्याद्री वाहिनी.
रिपीट हेडलाईन ह्या वापरायच्या. पुणे पुणे आणि नागपूर महानगर प्रादेशिक विकास प्राधिकरणाच्या हद्दीतल्या प्रकल्पांसाठी लागणाऱ्या जमिनींचं मूल्यांकन करण्यासाठी समिती नेमण्याचा निर्णय राज्य सरकारनं घेतला आहे. वर्षा निवासस्थानी झालेल्या बैठकीत मुख्यमंत्र्यांनी हा निर्णय घेतला असून काल त्यांनी पुणे आणि नागपूर महानगर प्रदेश विकास प्राधिकरणामार्फत सुरू असलेल्या विविध विकास कामांचा आढावा घेतला. विकास प्रकल्प पूर्ण करताना कालबद्ध मर्यादेत पूर्ण करावा, जेणेकरून नागरिकांना त्याचा लाभ मिळेल, असं मुख्यमंत्री यावेळी म्हणाले. पुणे महानगर प्रादेशिक विकास प्राधिकरणामार्फत 960 कोटी रुपयांचा शिलकीचा अर्थसंकल्प मांडण्यात आला असून एकूण निधीपैकी सुमारे 53% निधी मेट्रोसाठी खर्च करण्यात येत असल्याची माहिती प्राधिकरणाचे महानगर आयुक्त सुहास दिवसे यांनी दिली. नागपूर महानगर विकास प्राधिकरणाच्या कामांचं सादरीकरण महानगर आयुक्त शीतल उगले यांनी केलं. सध्या प्राधिकरणामार्फत सुमारे 1,500 कोटी रुपयांचा विकास प्रकल्प सुरू असून त्यात विकास कामांबरोबरच सौंदर्यीकरणावरही भर देण्यात येत आहे, असं त्यांनी सांगितलं. या बैठकीत उपमुख्यमंत्री अजित पवार, नगरविकास मंत्री एकनाथ शिंदे, नागपूरचे पालकमंत्री डॉक्टर नितीन राऊत, मुख्य सचिव संजय कुमार यांच्यासह अनेक अधिकारी उपस्थित होते. शासकीय अधिकारी आणि कर्मचाऱ्यांसाठी असणाऱ्या वैद्यकीय खर्च प्रतिपूर्तीमध्ये कोरोनाचा समावेश करायचा निर्णय काल राज्य सरकारनं घेतला. आरोग्यमंत्री राजेश टोपे यांनी ही माहिती दिली. शासकीय अधिकारी, कर्मचारी आणि त्यांच्या कुटुंबियांना आकस्मिक तसंच गंभीर आजारांवर खासगी रुग्णालयात उपचार घेतल्यास होणाऱ्या वैद्यकीय खर्चाची प्रतिपूर्ती दिली जाते. यासंबंधीच्या मार्च 2005च्या शासन निर्णयात हृदय आणि फुफ्फुसाशी निगडीत आजारांचा समावेश होता. मात्र कोरोना उपचारांच्या खर्चाबद्दल स्पष्टता आणण्यासाठी काल त्यात कोरोनाचाही समावेश करण्याचा निर्णय झाला. हा निर्णय 2 सप्टेंबरपासून पूर्वलक्षी प्रभावानं लागू होणार आहे. देशातलं पहिलं आंतरराष्ट्रीय क्रीडा विद्यापीठ महाराष्ट्रात स्थापन होत असून 2020-21च्या अर्थसंकल्पात या विद्यापीठाच्या स्थापनेची घोषणा करण्यात आली होती. नुकत्याच झालेल्या विधिमंडळ अधिवेशनात दोन्ही सदनांनी याला मंजुरी दिली. येत्या शैक्षणिक वर्षापासून या विद्यापीठात प्रवेश प्रक्रियेसह अभ्यासक्रम सुरू केला जाईल, अशी माहिती क्रीडा आणि युवक कल्याण मंत्री सुनील केदार यांनी काल मुंबईत वार्ताहरांशी बोलताना दिली. सध्या पुण्यातल्या बालेवाडीत शिवछत्रपती क्रीडा संकुलात विविध खेळांच्या आंतरराष्ट्रीय दर्जाच्या सुविधा उपलब्ध आहेत. हे लक्षात घेता नवं विद्यापीठ सध्या तिथेच कार्यान्वित करण्यात येणार आहे. या विद्यापीठासाठी स्वतंत्र इमारत बांधकामाला सुमारे 400 कोटींचा खर्च अपेक्षित असल्याचं केदार यांनी सांगितलं. विद्यापीठाच्या स्थापनेनंतर पहिल्या वर्षी क्रीडा विज्ञान, क्रीडा वैद्यकशास्त्र आणि क्रीडा व्यवस्थापन हे 3 अभ्यासक्रम सुरू केले जातील. याकरता प्रत्येक अभ्यासक्रमात 50 विद्यार्थ्यांना प्रवेश देण्यात येईल. त्यानंतर पुढच्या वर्षामध्ये मागणी आणि आवश्यकतेनुसार आणखी अभ्यासक्रम सुरू केले जातील. यातून क्रीडा प्रशिक्षणासाठी नोकरीच्या नवीन संधी उपलब्ध होतील, असं केदार यांनी सांगितलं.
हे क्रीडा आंतरराष्ट्रीय क्रीडा विद्यापीठ आपण स्थापन करणार राज्याचं, आणि ते आपण पुणे जिल्ह्यामध्ये बालेवाडी या ठिकाणी सुरू करतो आहे. कोरोनाचा उपक्रम चालू, कोरोनाचा फेज चालू, अशा याच्यामध्येही आपण त्याची पूर्ण जी काही प्रक्रिया असते ती प्रक्रिया आपण पूर्ण केली. त्या सगळ्यामध्ये सगळे सरकारी अधिकारी, कर्मचारी, स्पोर्ट्स विभागामध्ये काही आम्ही इतर विद्यापीठाचे रिटायर झालेले कुलगुरू यांचाही समावेश करून घेतला आणि अशा प्रकारे ते पूर्ण करून रूपास आणलेलं आहे.
राज्यस्तरीय ऑनलाईन महारोजगार कार्यक्रम येत्या 20 डिसेंबरपर्यंत सुरू राहणार असल्याची माहिती कौशल्य विकास आणि उद्योजकता मंत्री नवाब मलिक यांनी दिली आहे. कौशल्य विकास, रोजगार आणि उद्योजकता विभागानं 12 आणि 13 डिसेंबरला असा मेळावा घेतला होता. या मेळाव्याला उद्योजक आणि उमेदवारांकडून मिळालेल्या प्रचंड प्रतिसादामुळे त्याला मुदतवाढ देण्याचा निर्णय झाला. रोजगार मेळाव्यात सहभागी होण्यासाठी इच्छुक तरुणांनी rozgar.mahaswayam.gov.in या संकेतस्थळावर मोबाईल क्रमांक, ईमेल ID आणि आधार क्रमांकासह नोंदणी करायची आहे. तसंच कुशल उमेदवारांच्या शोधात असलेल्या उद्योजकांनीही या संकेतस्थळावर नोंदणी करावी, असं आवाहन मलिक यांनी केलं आहे. कोकणात पर्यटन विकासाला चालना देण्यासाठी बीच शॅक धोरणाला मंजुरी देण्यात आली आहे. कोकणात पंचतारांकित हॉटेल्स गुंतवणूक करत आहेत. यापुढील काळातही कोकणाच्या पर्यटन विकासासाठी विविध क्षेत्रांच्या अनुषंगानं व्यापक कार्य केलं जाईल, असं पर्यटन मंत्री आदित्य ठाकरे यांनी काल सांगितलं. कोकण आणि औरंगाबाद जिल्ह्यात पर्यटनाला चालना देणाऱ्या विविध उपक्रमांच्या आढावा बैठकीत ते काल बोलत होते. या बैठकीत महसूल आणि ग्रामविकास विभागाचे राज्यमंत्री अब्दुल सत्तार, सिंधुदुर्ग जिल्ह्याचे पालकमंत्री उदय सामंत, खासदार विनायक राऊत यांच्यासह अनेक जण उपस्थित होते. कोकणात पर्यटन क्षेत्रात बीच शॅक टुरिझम, टेंट टुरिझम, कॅराव्हॅन टुरिझम यासह कृषी पर्यटनालाही मोठा वाव आहे. होमस्टेच्या माध्यमातून इथं स्थानिक नागरिकांना मोठ्या प्रमाणात रोजगार मिळू शकतो. कोकणात पर्यटनाला चालना देण्यासाठी मुंबईपासून गोव्यापर्यंतच्या विविध पर्यटन ठिकाणांच्या विकासाचा आराखडा तयार करून त्याला चालना देण्यात येईल, असंही आदित्य ठाकरे यांनी यावेळी सांगितलं. त्याचबरोबर औरंगाबाद जिल्ह्यातल्या सर्व पर्यटन स्थळांना जोडणाऱ्या रस्त्यांचं जाळं मजबूत असावं, रस्ते दर्जेदार कसे होतील या दृष्टीनं कार्यवाही करावी अशा सूचनाही त्यांनी दिल्या. पर्यटकांसाठी शहरात सुरू करायच्या हॉप ऑन हॉप ऑफ वाहतूक व्यवस्थेचा आढावाही त्यांनी घेतला. औरंगाबाद जिल्ह्यातल्या अजिंठा आणि वेरूळ हे दोन्ही जागतिक स्तरावरची पर्यटन स्थळं आहेत. औरंगाबाद, फरदापूर, अजिंठा लेणी यासह जिल्ह्यातल्या सर्व पर्यटन स्थळांना जोडणाऱ्या रस्त्यांचा दर्जेदार विकास आवश्यक आहे. यासाठी सार्वजनिक बांधकाम विभाग, जिल्हा परिषद आदी संबंधित यंत्रणांनी समन्वयानं काम करावं अशा सूचना अब्दुल सत्तार यांनी या बैठकीत दिल्या. लांब पल्ल्याच्या रेल्वेगाड्यांनी प्रवास करणाऱ्या प्रवाशांना मुंबई क्षेत्रातल्या जवळच्या संबंधित स्थानकापर्यंत जाण्यासाठी उपनगरीय रेल्वेचा प्रवास करायला रेल्वे मंत्रालयानं काल परवानगी दिली. राज्य सरकारच्या विनंतीवरून अशी परवानगी देण्यात येत असली तरी त्यासाठी प्रवाशांना काही अटींची पूर्तता करावी लागणार आहे. मुंबईहून बाहेरगावी जाणाऱ्या गाडीचं आरक्षित कन्फर्म तिकीट असणाऱ्या प्रवाशांनाच त्या गाडीत चढण्याच्या स्थानकापर्यंत पोहोचण्यासाठी लोकलनं प्रवास करता येणार आहे. तर परगावाहून मुंबईत उतरलेल्या प्रवाशांना त्यांच्या मुक्कामी पोहोचण्यासाठी असा प्रवास करायला परवानगी असणार आहे. या दोन्ही बाबतीत लोकलचं एकेरी तिकीट काढायलाच परवानगी असणार आहे. राज्यातल्या लॉकडाऊनमुळे 1ली ते 8वी पर्यंतच्या शाळा अजूनही बंद आहेत. ग्रामीण भागही त्याला अपवाद नाहीत. त्यात ऑनलाईन शिक्षण फारसं प्रभावी पद्धतीनं होताना दिसत नाही. ही अडचण लक्षात घेऊन अमरावती जिल्ह्यात शाळा विद्यार्थ्यांच्या अंगणात हा उपक्रम राबवण्यात येत आहे. पाहूया त्याचा वृत्तांत.
कोरोना संसर्गामुळे राज्यात अजूनही 1ली ते 8वी पर्यंतच्या शाळा बंद आहेत. त्यामुळे ऑनलाईन पद्धतीनं शिक्षण देण्यावर भर दिला जातोय. ग्रामीण भागात मात्र विद्यार्थ्यांना ऑनलाईन शिक्षणासाठी अनेक अडचणींना सामोरं जावं लागत आहे. या परिस्थितीवर मात करण्यासाठी अमरावती जिल्ह्यातल्या शिक्षकांनी गावातल्या भिंती बोलक्या करून शाळा विद्यार्थ्यांच्या अंगणात हा अनोखा उपक्रम सुरू केला आहे. चांदूर रेल्वे तालुक्यातील 67 गावात हा उपक्रम राबवला जातोय. गावात तसंच अंगणवाडी तसंच किराणा दुकानाच्या भिंतींवर शैक्षणिक अभ्यासक्रमातली भित्तिचित्र लावून संपूर्ण गावाला चालत्या बोलत्या शाळेचं स्वरूप प्राप्त झालं आहे.
मुलांच्या दारी शिक्षण हा उपक्रम शिक्षकांनी मुख्याध्यापकांनी राबवलेला आहे. ज्यामुळे विद्यार्थी जे व्हॉट्सअप मोबाईलद्वारे शिक्षण घेत होते, ऑनलाईन शिक्षण, परंतु प्रत्येकच पालकाजवळ अँड्रॉइड मोबाईल नाही आणि असला तरी पण तो पालकाजवळ असतो. त्याच्यामुळे पुरेसा उपयोगी विद्यार्थ्यांना होत नाही. त्यामुळे इंग्रजीचे गाव, मराठीचे गाव आणि विज्ञानाचे गाव याप्रमाणे तिन्ही प्रकारचे चार्ट ज्या ठिकाणी विद्यार्थी खेळतात त्या ठिकाणी लावल्या गेलेले आहेत. त्याचा उपयोग पालक आणि विद्यार्थी या दोन्हींना होत आहे आणि खऱ्या अर्थाने मुलांना शिक्षण मिळत आहे.
पुस्तकांपेक्षा भित्तिचित्राच्या माध्यमातून शिक्षण घेताना विद्यार्थी अधिक उत्साहानं शिकत आहेत, असं जिल्हा परिषदेचे मुख्याध्यापक राजेंद्र वानखेडे यांनी सांगितलं.
भाषा, गणित, विज्ञान, सामाजिक शास्त्र चांदूर रेल्वे पंचायत समितीचे आदरणीय गट शिक्षणाधिकारी राजनगर साहेब यांनी हे उत्कृष्टरित्या संपूर्ण तालुक्यामध्ये राबवित आहे आणि या उपक्रमाचा विद्यार्थ्यांना भरपूर फायदा होत आहे.
आमच्या सरांनी हे वाले चार्ट लावले आहे जे आमच्यासाठी आहे. ते आम्ही इथे खेळत असतो तर ते आम्हाला इकडं आमचं लक्ष जाते तर आम्ही अभ्यास पण करू शकतो. कोरोनामुळे शाळा बंद असल्याने सरांनी आमचा अभ्यास व्हावा म्हणून आमच्या इथे चार्ट लावलेले आहे आणि त्याचे फायदे आम्हाला पण आहे आणि जे इतर छोटे मुलं आहेत त्यांना पण आहे.
गावातील शिक्षण मित्रही या उपक्रमात विद्यार्थ्यांना शिक्षण देण्यासाठी मदत करत आहेत. ग्रामीण भागातील विद्यार्थी शिक्षणापासून वंचित राहू नये यासाठी राबवण्यात येत असलेला हा उपक्रम नक्कीच स्तुत्य आहे. यशपाल वरठे, दूरदर्शन बातम्यांसाठी, यवतमाळ.
देशभरात कोरोना संक्रमणाचा प्रादुर्भाव अजूनही ओसरलेला नसला तरी हळूहळू दैनंदिन व्यवहार टप्प्याटप्प्यानं सुरळीत होत आहेत. अशावेळी कोरोनापासून बचाव करायचा असेल तर हात स्वच्छ धुणं, परस्परांपासून सुरक्षित अंतर राखणं आणि मास्क व्यवस्थित लावणं या सुरक्षा उपायांचं जबाबदारीनं पालन करणं अतिशय गरजेचं आहे. त्यामुळे मास्क लावा आणि सुरक्षित राहा.
जागतिक महामारी कोविड-19 ज्याच्या विळख्यात संपूर्ण मानवजात सापडली आहे, त्यावर लस किंवा परिणामकारक औषधाचा शोध सुरू आहे. मात्र तोपर्यंत खबरदारी आणि प्रतिबंध हेच उपाय आहेत. आता अनलॉक 5 अंतर्गत संपूर्ण देशात व्यवहार सुरू झाले आहेत. आता आपण अधिक सतर्क राहणं गरजेचं आहे. कोविड-19 ला आळा घालण्यासाठी मास्क हा सर्वात प्रभावी उपाय असल्याचं आढळून आलं आहे.
ऑस्ट्रेलिया क्रिकेट सामन्याच्या दुसऱ्या दिवशी आज भारताचा पहिला डाव सर्व गडी बाद 244 धावांवर संपुष्टात आला. ॲडलेड इथं सुरू असलेल्या या सामन्यात काल पहिल्या दिवशी भारतानं 6 बाद 233 इतकी धावसंख्या उभारली होती. मात्र आज भारतीय संघातल्या अखेरच्या फळीच्या फलंदाजांना यात केवळ 11 धावांची भर टाकता आली. आर अश्विन पहिल्याच षटकात 15 धावा काढून बाद झाला. इमान साहा मिचेल स्टार्कच्या चेंडूवर तंबूत परतला. उमेश यादवही चमक दाखवू शकला नाही. ऑस्ट्रेलियाच्या पहिल्या डावाला नुकतीच सुरुवात झाली आहे. हवामान. राज्यातल्या बहुतांश भागात थंडीचा जोर वाढला आहे, तर काही भागात किमान तापमान सरासरीपेक्षा अधिक आहे. राज्यात गडचिरोली, अहमदनगर शहर आणि परिसर, जालना, नांदेड, परभणी आणि ठाणे जिल्ह्यात थंडीच्या प्रमाणात वाढ झाली आहे. येत्या 24 तासात राज्यातलं हवामान प्रामुख्यानं कोरडं राहील असा अंदाज हवामान विभागानं वर्तवला आहे. दूरदर्शनच्या सह्याद्री वाहिनीवरच्या आपल्या बातम्यांचं थेट प्रसारण आपण DD सह्याद्री न्यूज या यूट्यूब चॅनलवर पाहू शकता. तसंच ताज्या बातम्या जाणून घेण्यासाठी @DD सह्याद्री न्यूज या ट्विटर हँडलला जरूर फॉलो करा. शेवटी ठळक बातम्या पुन्हा एकदा. केंद्र सरकारनं शेतकऱ्यांच्या हितासाठी कृषी कायद्यात केलेल्या सुधारणा. भारतीय कृषी क्षेत्रात एका नवीन अध्यायाची सुरुवात. केंद्रीय कृषी मंत्र्यांचं शेतकऱ्यांना पत्राद्वारे आश्वासन. शेतकरी बंधू भगिनींशी खुल्या मनानं संवाद साधणारं हे पत्र शेतकऱ्यांनी आणि देशवासियांनी अवश्य वाचावं, पंतप्रधान नरेंद्र मोदी यांचं आवाहन. शेतकऱ्यांना आपल्या हक्कांसाठी आंदोलन करण्याचा अधिकार असला तरी यामुळे इतरांचं जीवन विस्कळीत होणार नाही याची खबरदारी घ्यावी, सर्वोच्च न्यायालयाची स्पष्टोक्ती. लक्षावधी देशवासियांचं भरणपोषण करणारा शेती हा एकमेव व्यवसाय कोरोना काळात अर्थव्यवस्थेला आधार देत होता. उपराष्ट्रपतींकडून कृषी क्षेत्राचं कौतुक. देशांतर्गत उद्योग क्षेत्रातून 27,000 कोटी रुपयांची संरक्षण सामग्रीची खरेदी करायला केंद्र सरकारची परवानगी. देशात उपचाराधीन रुग्णांचं प्रमाण कमी होऊन एकूण रुग्णसंख्येच्या 3.14% आणि रुग्ण बरे करण्याचा राज्य सरकारचा निर्णय. देशाचं पहिलं आंतरराष्ट्रीय क्रीडा विद्यापीठ महाराष्ट्रात. येत्या शैक्षणिक वर्षापासून प्रवेश प्रक्रिया सुरू. आणि ॲडलेड इथल्या पहिल्या क्रिकेट कसोटी सामन्यात आज दुसऱ्या दिवशी भारतीय संघाचा पहिला डाव 244 धावात संपुष्टात. याबरोबरच हे बातमीपत्र संपलं. पुन्हा भेटूया दुपारी 1 वाजता पुढच्या बातमीपत्रात आपल्या सह्याद्री वाहिनीवर. कोरोनापासून बचाव करण्यासाठी सुरक्षित अंतर राखा, हात स्वच्छ धुवा आणि मास्क व्यवस्थित लावा. नमस्कार.